with that, thank you once again. I'll close this session and hand over to Dr. Aurora to start our second session.
Thank you.
Thank you, Dr. Laura, and thank you, Dr. Nandi, for the wonderful session handled. Now we would move to the next session. That would be moderated by Professor Arora. Professor Arora has already been introduced by Dr. Awasthi, so I'll not take much of the time and hand over the session to Dr. Arora, please. So, for you to take over, thank you.
Thank you. Thank you, Gurleen. You can remove the slide, please.
Sure, sir. Thanks.
Okay. Thank you very much. And I'd like to start the next session. It was very nice to have Dipankar talking about. Gurleen, can you mute your camera? Thank you very much. So we had a very nice session from Dipankar. Normally we don't discuss thymus so elaborately. So I think it was very good to listen to Dipankar, who gave us a very nice overview of these, thymus and its atrophy and everything. So now what we want to do is to move on to the next speaker, and in line we have Dr. Himanshu. Himanshu Kumar is going to talk about innate components of the immune system, and I'm very happy to introduce him because he's a young speaker, young scientist, and doing very good immunology. He works as associate professor at the Department of Biological Sciences in Indian Institute of Science, Education and Research at Bhopal. He earned his master's degree from Lucknow University and then got a fellowship for PhD to be completed in Osaka University, Japan. He moved on to have the postdoctoral fellowship in Japan itself, and then came back to India and joined ICER Bhopal in 2010. Kumar is also recipient of Ramanujan National Fellowship of DST in 2010. He is adjunct associate professor of immunology, Frontier Research Center, a top immunology institute in the world. Last year, he was appointed as editor-in-chief of International Reviews of Immunology. He is member of National Academy of Medical Sciences. His research interests, they are innate immunobiology. His major focus is transcriptional and translational level regulation of antiviral innate immune responses during flu and or another virus infections. So I hand over to Dr. Himanshu, who will speak on innate components of the immune system. Himanshu, please. Dr. Himanshu, can you start your lecture?
Yes. So first of all, am I audible? I want to cross-check.
Yes.
Okay, that's great. So can you see my slide?
Yes, they are almost there. Yes. Yeah, we can see that.
Okay, that sounds great. So first of all, I would like to thank the organizer for giving me this opportunity to take a class or a session on innate component of immune system, and very much thank to Professor Arora for very nice introduction. And so it's a good that the session is a bit early, so I have a little liberty that if my talk will spill over a little over the time, then I think you don't mind so much. So I have designed this talk for.
I suggest you to leave some time for discussion, because as you see,
Sure. I will definitely do it. I will finish within a time, but if something go over, then I think it is. I will try to finish in the time. So Yeah. So basically, I have designed this talk for more for students, and initially I thought that I will make a more research oriented, but then I thought no, it's we should since it is an immunology course, I should move slowly to the complexity, and then I will end my talk with the research. So let's start it. So the title of my talk is Innate Component of Immune System, and this slide, if you see, can you see next slide? Yeah. So if you see this slide, you can make out this is a scanning electron micrograph of large number of bacteria in gums in human mouth, and you can understand that if there are so much bacteria, then what is our identity? So I would like to start my talk with one very simple question, that our human body is really human. So here, what I want to precisely say that I want to show the importance of innate immunity that generally we lack in several talks or in articles, right? So I thought I should start with like that. So my simple question is: are human bodies real human? So if you look at the previous slide, then I think you will convince with me that this answer is no. And why it is no? Because previously in 1972, people attempted to find out the number of microbes compared to the human cell, and they found out that this number of microbes are 10 times more than the human cell. And since this was a reasonably old work, so this was revisited, and then people try to find out. And if we take this 10 times, then it will constitute only 9.1% human cell, and rest are the microbes. So after revisit in 2014, this was published in Cell, and they found out that the number of bacteria is about 1.3 times more than the human cell. Although this in this study they have excluded the fungi, viruses, and phages, and this if we take 1.3 times, still the number of microbes are more, and human cells are 43.5%. And if we consider at least an order of magnitude, this fungi, viruses, and phages, then again it will come around the same number, about 9.5. So here, just I wanted to say that our body is more dominated by the microbes, and this is all about the game of size and number. And so next, how these microbes are interacting with the host. So there are several ways by which they are interacting. I am showing a very simple way. So microbes, so there could be a commensal association where microbe is neither benefited or losing anything, but host is of course fine. There is a mutualism. So you know that there are several microbes which are good for our health, and they are present in symbiotic association. They benefit each other. This is important because now we understood this importance of mutualism, and then now we are using the probiotics when we have a stomach upset. But the last part is the parasitism. Of course, there are so many microbes, or I will say reasonably less number of microbes which cause the disease. And this kind of association, we call it as a parasitism. It could be so when I say microbe, it includes all microbe, bacteria, virus, fungus, and so on. So if you look at the number, after number, if you see the number of genes provided by these microbes, then you will be really surprised, and it is about 200 times more than the host gene. So we have about 23,000 to 30,000 gene, and the gene which is given to the host is approximately 200 times more. So you can understand the stress on the human cell or human system. So the human body or human immune system need to handle so many genetic pressure. So in addition, or furthermore, if you see the innate immunity, although it is evolved quite early, about 700 million years ago. But the evolution of microbe, if you notice, the microbes are evolving much more rapidly, and still many host restriction factors are undergo positive selection in order to tackle over these microbes. So collectively, I can say that outnumber of microbes. So this is one thing. Another is genetic or gene pool derived from the microbes, and faster evolution of microbe results to the development of innate immunity as a first line of defense against the plethora of microbes. So here you should understand that every moment, every time we are exposed with numerous microbes, so many microbes, but our efficient innate immune system, they keep all these microbes away, and we don't fall sick. We fall sick very rarely, right? We don't fall sick every day, every moment. So this is all about the innate immune system. So our innate immune systems are working very efficiently. That's why we don't fall sick. Only when there is some severe pathogen comes in contact with the host, then it causes the disease. So let me explain the components of innate immunity. So the components of innate immunity is a physical barrier. We know very well the physical barrier. In subsequent slide, I will show what are the physical barriers and biochemical barriers. There are some chemicals, or there are some biochemical reaction metabolites are produced, which is keeping the microbes away, or we are not infecting by those microbes. And there is a microbiological barrier. So there are some commensal bacteria or which take the shelter and food from human body, and in return they produce some metabolite which inhibits the pathogenic microbe. So this is one of very important barrier. And the most extensively studied the barrier is a humoral component of innate immunity. So humoral is all those soluble component which is produced by the host. For example, antimicrobial peptide, proteins like MBP, mannose binding protein, CRP, C-reactive protein, and so on and so forth. And some enzymes like lysozymes. So they are all soluble form, and they keep the microbe away from the host, or they do not allow the microbe to grow in more number in order to cause any problem to the host. So this is one of humoral component. Another is the complement, and this complement is a family of protein which is synthesized by liver, and they have basically three major functions. They opsonize, so they can bind to the target microbe, and then it will be readily taken up by the phagocytic cells. Another is they also induce the inflammation. So you probably remember that there are a small component of complement like C5a and C3a. All those components induces the inflammation. I will tell how inflammation is important in subsequent slide. And so complements are playing a very important role, and it's a humoral component. And I strongly believe that Dr. Arvind Sahu, he will talk more detail after this talk about the complements. And today I will talk about the microRNA against viruses. So these are also some kind of response. So when there is a virus infection, there are some microRNA which is induced, and they directly bind to the virus and they control the viral infection. So I will talk more about this in subsequent slide. And the final component is the cellular component. So cellular component is there are variety of cells. This includes non-immune cells as well. So if there is a situation, then the non-immune cell like epithelial cell, they can sense the microbial pathogen, and they can induce very nice response in order to control the infection. Okay, and these cellular components basically expresses very well known. We all are aware about the pattern recognition receptor, which basically sense the metabolic pathways unique to the microbe, and this cellular component plays a very important role against the variety of bacterial, viral infection. So let me start with physical barrier. So here you can see that there are variety of things which keep the microbes away. It's not only microbe, other than microbe, abiotic stuff like a pollutant and all those things. These things can be taken care by the like cilia, mucus, and cough reflex. So mucus, which is produced in gastrointestinal tract, acid produced in our stomach. We have a HCl, which is required for digestion. So they also control the microbe as well. The intestinal enzymes, the colonic flora. So here, this is a very good example of microbial barrier. If there is a disturbance in this flora, then we feel this stomach upset and other problems. So intact skin, of course, is skin, which is the barrier between the environment and our internal environment. So they keep the control, and the sweat which is produced, and the sebum which is produced from the skin. So all these things basically constitute a very strong physical and biochemical barrier, and we are protected from most of the microbial infection. So there are some key players of cellular players of innate immunity. I will just show a few cells like neutrophils, and I think about neutrophils, Dr. Moni Deepa will talk in more detail in today's other session. And so these innate immune cells are basically playing a very important role in, again, not only microbial infection, some parasite infection and other allergy-like responses, natural killer cells, which plays a very important role against viruses. There are active immune cells. So you will have a lot of talk about the B lymphocyte and T lymphocyte in great detail. So they are the adaptive immune cells, and these cells are playing a very important role in development of specific immunity against the microbe. If the microbe is not cleared by the innate immune system, then all this information is supplied to the adaptive immunity along with all microenvironment, and this microenvironment will consist of macrophages and dendritic cell and some inflammatory cytokine, and this thing will be all this microenvironment activate antigen specific or microbe specific B and T lymphocyte responses. Macrophages and DCs are present in between the innate and adaptive immunity. And how? Because macrophages can induce they can sense the pathogen. They can also express the MHC class 2 molecule, and they can present the antigen. So in that way, the macrophages and DCs are sitting just in between the innate and adaptive immunity. So if you see the cellular component, they are very important, and they are bridging between the innate and adaptive immunity. Complements are the humoral component. They are also bridging the innate and adaptive immunity. You know that antibody-mediated complement fixation and all those things, right? So how this cellular component works? It's a very simple way. The cellular component, they basically express the pattern recognition receptor, which senses the pathogen-associated molecular pattern. And regarding pattern recognition receptor, Dr. Praful Kumar Taylor will talk in great detail. And these PRRs basically sense the PAMs, and when they sense the PAM, then they initiate inflammation. Initiate inflammation means they will synthesize some innate inflammatory cytokine like IL-6, TNF, and so on and so forth. And it will also activate the innate immune responses. Here, what I want to say that innate immune responses, when these cells are treated with inflammatory cytokine, the innate immune cells, then their capacity is enhanced to manifold. For example, macrophage if treated with some inflammatory cytokine, then their phagocytic activity is enhanced to manifold. DC's activities are also enhanced manifold. They can produce ROS and all those things to manifold high. They can much more efficiently clear the microbe. In addition to initiate inflammation and activate innate immune response during virus infection, this induces the type 1 and type 3 interferon. So type 1 and type 3 interferon, I will explain more about the function of these inflammatory cytokine as well as type 1 interferon in subsequent slide in more detail. And most of microbe, please note that most of microbe are cleared at this stage. If it is not cleared, it is making a bed or making a condition for the development of pathogen-specific adaptive immunity, which is triggered by the B and T lymphocyte. Okay, so there are variety of inflammatory mediators. It's not only the cytokines, and it's not only the pro-inflammatory cytokines. There are several mediators. It could be a vasoactive amine, it could be a histamine and serotonin, it could be escanoids. We know very well there are thromboxane, leucine, and prostaglandins, which is synthesized from arachidonic acid by the action of cyclooxygenase, and they also induce the inflammation. There are peptides like a bradykinin, and there are proteins. You know proteins very well, like complement component C3A, C5A, and there are inflammatory cytokines like IL-1 beta, IL-6, and TNF. So basically, these are the inflammatory mediators. On another hand, the type 1 and type 3 interferons. So type 1 interferon is basically consists of 13 member alpha, and there is a single interferon beta, and there is a type 1 interferon inducible gene. So when you treat the cells with interferon, type 1 interferon, then this triggers expression of hundreds of genes, and all those genes collectively create an antiviral state. You know that viruses hijack the host cellular machinery, and then they dictate the cells, the host cell, to make their own protein and nucleic acid. So this type 1 interferon and type 1 interferon inducible basically cause the antiviral state by, for example, inhibiting the translation, by protecting the virally infected cell from other viruses, and so on and so forth. Okay, and there is a type 3 interferon, which is not recently, yeah, but more recently it was found out that there are type 3 interferon, which is consist of 4 members, lambda 1, lambda 2, and lambda 4. So all these type 1 and type 3 interferon creates an antiviral state. And how this type 1 and type 3 interferon works, so you can see that this type 1 and type 3 interferon, they can create apoptosis in virally infected cell. So they basically, because we cannot clear the virus, it's better to kill the cell rather than killing the virus. Okay, so they can induce apoptosis. They can make healthy cells resistant from the virus infection, as you can see in this cartoon. They also depress the cellular synthesis pathway, because now virus hijack the system. So this system should not run so fast. If it will run so fast, then they will make much more virus. So basically, type 1 interferon depresses the cellular synthesis pathway or metabolic pathway in general, and they prepare and activate the professional immune cell for development of virus-specific immunity. So, since several viruses cannot be cleared, then this needs a much more advanced level of defense system, that is adaptive development of appropriate adaptive immunity. Okay. So how pro-inflammatory cytokine works? So pro-inflammatory cytokine is just very important. It gives an alert system. And when there will be alert system, okay, there is a virus infection, all systems should be alert. So all professional guys, all professional immune cell will come at the site of infection, and they try to clear it. So this is a scenario when there is a virus infection, then when there is a no type 1 interferon, then virus may dominate over the innate over the host. Okay. But once this system is treated with the type 1 interferon, here you can see that, then this heightened immunity can easily manage the virus infection. Okay. So however, all these responses are very important against the virus infection, but all these things need to be extremely tightly regulated. If it is not regulated, then everything will go hayway, and then that leads to the autoimmune disease. Towards end, I will show that if there is some dysregulation of some microRNA, then that leads to the autoimmune disease. However, if these microRNAs are enhanced, then this will be a very good, this will provide a very good protective response against virus infection. I will show you towards end. And this about regulation of innate immunity, Dr. Osamu Takeuchi from Osaka University, he will talk today, later. So now you can see by looking at this picture the importance of regulation. So this is a system. If the system is not regulated, then everything will be chaos. However, if the system is regulated and perfectly work, then that leads to the better performance. So this is just a social example of regulation. So similar thing is needed in the cells. So therefore, we have a variety of regulation. Transcriptional level regulation. Now we know there are so many kinds of RNA that regulates the pathway: circular RNA, long non-coding RNA, microRNA, and now there are some transcriptome thing. So there is some modification, and that causes a regulation. Then translational level regulation. There are so many levels of regulation, and we need all those regulation. And if something goes wrong in regulation, then you remember the previous image. There will be a chaos. Okay. So this is a pattern recognition receptor. This is a very simplified image of pattern recognition receptor. You can see that there are so our system, our cell has a very beautiful defense system or recognition system. We have a very nice sensors over the cell surface. We have a nice sensors over the endosome, and we have a sensors in the cytoplasm. If something will escape from this membrane and endosome, then there will be, which is the case for the viruses, most of viruses, RNA viruses. So then there will be a sensors. These are the RNA sensors, which senses RNA molecule from viruses. There is a sensors for DNA. They're like IFI16 and sting. Sting is one of the key molecule which transduce downstream signal. Okay. So we have a cytosolic sensor. Then we have a sensor which can sense variety of molecules. We know that inflammasome and NLRP3 inflammasome is very well studied. And if you look at the ligands for inflammasome and NLRP3 inflammasome, it's a huge. So all these things induces the synthesis of IL-1 beta, which is very tightly regulated. Okay. And there are M2 sensors, which senses the DNA molecule. So for more detail, I think later on, Dr. Praful Kumar Taylor will talk in great detail about this pattern recognition receptor. So I will not take further more on this PRR. So now I will talk about our work. Which I will try to present in very simplified way. So this, we had a question. We wanted to find out a microRNA which can regulate the expression of regi, and accidentally or luckily we could find out that this microRNA is not only playing a very important role in regulation of regi expression, but it is playing a important role in controlling the influenza virus that I will demonstrate in subsequently. So when there is a virus infection, this is an RNA virus. I have taken influenza virus as well as NDV and several other viruses we have used. So when there is an infection, this genome is transported inside the cell, and over there is a regi. This regi will sense this molecule, and then this will induce the inflammatory cytokine and type I interferon. This is a very simple way, okay? And it will induce the antiviral responses or antiviral state. So we wanted to find out which microRNA can regulate this regi expression. So we have performed a lot of bioinformatic analysis, and finally we found out that there is a microRNA 485. We have performed bioinformatic as well as wet experiment, and we found out that microRNA 485 is a one of candidate among several, which plays a very important role, and this is induced in human lung, small alveolar lung epithelial cell, this lung cancer cell line A549. And so we found out that this microRNA is induced upon the virus infection, influenza as well as NDV, in fact, when we introduce the poly-IC, then it induces the expression of this microRNA. And as usual, this is very nice. This can bind to the regi, and this will reduce the type I interferon and inflammatory cytokine. And when this will happen, then if you infect, if you have a cell which is overexpressing microRNA 485, then the viral load will increase, right? So this is interacting perfectly. We have performed a lot of experiment, and then we showed that this is perfectly binding with this microRNA, and then this is reducing the expression of regi, and then there will be an increase in viral load. Here you can see this. This we have used NDV at 24 hours and like that. We have also used antiviral, and we have demonstrated this thing that this microRNA is playing a very important role. So we have performed similar experiment with influenza virus, and then we found out very interesting result. What we have found that initially, as per this hypothesis, the viral load was enhanced. But when we left the cell for longer time period, then we found out that viral load was reduced very much significantly, and cell looked much more healthy. So this was a quite interesting phenomena. We thought we have done some wrong in experiment. We have repeated this multiple times. I think maybe 10 times we have repeated, but we were getting the similar result. We have proved, we have seen by increasing the more viral load. So when we increase the viral load, the cell is very, very healthy. So then what we have done, we have tried to ask this question, how this is happening. So this is again the simple way. There will be a type I interferon and inflammatory cytokine, and when this inflammatory cytokine bind to the interferon receptor, and it will again induce the this regi. So regi is the inducible gene. Okay, so this will also induce the microRNA 485, and this microRNA 485 can bind to the regi. So far so good. I have explained in previous slide. Then we have analyzed the genome of this influenza virus, and we found out that, very interestingly, this microRNA is specifically targeting one of very important gene of the influenza virus, which encode for PB1. And please remember, PB1 is one of subunit of RNA polymerase of this virus. This polymerase is needed for the replication of virus. So this microRNA is specifically targeting this influenza virus PB1 gene. And if you see carefully, you can see that this is highly conserved in all recent as well as in very old strains. Okay, so this is very specifically binding. So what is happening when, and you know that when virus infect the cell, they can replicate much more faster. So in this scenario, what is happening? The genome is replicating much more faster, and since cellular cell has a limited number of microRNA, so this will bind more to the genome of influenza virus or gene of PB1. And if this will happen, then this will lead to the degradation of the genome, and then this will reduce the viral load. Okay, so we have demonstrated all this thing by creating this influenza virus by reverse genetic system. We have we got it from the Ron Fauché, and we created this virus, we mutated, and all those things, and we could very nicely demonstrated this these results as it is shown in this figure. So overall, what I can say that this so this is targeting the genome, and it is less available for the regi. So regi will be much more free, and then it will be it is acting like a double-edged sword against the influenza virus. So in that way, this microRNA is playing a very important role against the influenza virus infection, this control. And please remember, this microRNA is binding to the highly conserved region. And the unique property of influenza virus is variation, and most of variation is taking place in the HA and NA protein and other genes. But the virus cannot make a mutation in these regions, because this is an active site region. this, if they will make a mutation, then they cannot replicate efficiently. Okay. So, so next we have another study where we have shown that this microRNA 324, which is again targeting the PB1, is playing a very important role in controlling influenza virus, and flu basically inhibit this microRNA. Again, I will show the schematic. So when there is a virus infection, there will be a in cellular pool, there will be a microRNA 324. We have performed a lot of analysis, and then we discovered this microRNA. And we have shown that this is specifically binding to the PB1, okay? And when this microRNA is binding to the PB1, then this reduces the viral load. Again, in the similar phenomena as I discussed in previous slide or previous work. This microRNA can also bind to the CUEDC, which is a negative regulator of JAK-STAT pathway. And when it will bind to the negative regulator, then there will be a much more type I interferon. And then in that way, it will again act as, it will reduce the viral load. However, this influenza virus is a much more smart. What this guy is doing, this is basically suppressing the microRNA 324. Okay. And when it is suppressing the microRNA 324, it is basically evading the immune system. Okay. And another interesting thing is that this microRNA, which is binding to the influenza virus, is creating a mutation in new strains or several strains. You can see that this is, the amino acid is not changed, but it creates a mutation. Okay. And this mutation doesn't change the amino acid sequence. So this is a very good viral evasion strategy. Okay. So overall, what I can say that, but still this microRNA act as a antiviral factor. How? Because they are binding through the negative regulator of this JAK-STAT pathway. And if we can introduce the stabilized form of microRNA 324, we can use it as a therapeutic, not only against influenza virus, maybe we can use with for influenza virus, but we can also use for other viruses where the type I interferon production is very less. Okay. So this is since I have presented in very abstract way. So if you are more interested to talk or to understand the biology of microRNA 485 and microRNA 324, then you can go through this publication. So overall, microRNA could be a very major game changer. And now there are several microRNA which is in various clinical trial, and if we clinical trial for other disease, and probably this microRNA can be useful for taking it to the clinic, because now the microbes, or including virus, they are developing the drug resistance like Tamiflu resistant influenza virus and so on and so forth. So we need to change the strategy to tackle over these viruses. However, these studies need to further done in the in vivo condition. I don't have those situations, so I cannot do it currently. So this is another study where we have discovered the microRNA 30E, and this microRNA 30E is induced through the PAMP or DAMP signaling along with pro-inflammatory cytokine and type I interferon. And this microRNA targets the negative regulators, most of negative regulator of this PRR mediated signaling pathway. And if there is a dysregulation of this microRNA, then this can lead to the autoimmune disease, because there will be a much more production of type I interferon and inflammatory cytokine. And we have demonstrated using the mouse model, if we give this stabilized form of stabilized form of this microRNA to the mouse, then we can reduce this SLE signature in the mouse. So this is our recent work, and if you are more interested in this study, then you can go through this thing. And with that, I would like to thank my collaborators, Ashwin Raut, who provided this BSL-3 facility, my mentor, Professor Shizu Akira, he is my PhD supervisor, and Professor Osama Takeuchi must be knowing. He taught me, in fact, Professor Osama Takeuchi taught me how to create the knockout mice. I have created IPS-1 knockout mice. Akinori Takaoka, he has a huge expertise in type I and type III interferon. Ron Fosche, he provided the reverse genetic system. Peter, please, he provided the GFP labeled NDV, and there are so many funding agencies, and this is my vibrant group. We all work together in order to make some good story and important discovery in the field of antivirus. With that, I will thank, and now I'm happy to take any question. I believe I have done in, okay, I have exceeded a little more, sorry.
Thank you very much. Thank you, Imanshu. It was a good overview. Initially, you took over all the innate immune components. You also introduced to many upcoming talks. That was very nice of you. And it was great to see some of your research work regarding microRNA and control over viral infection. So before I open, there are 4 or 5 questions in the question answer box. I'll ask some of those to you. But my question is that since you showed that there is a microRNA which can be used to regulate viral replication as well as interferon type I interferon production, is there? Have you tried some of its effect on COVID-2, which is the recent
Yeah, that's a very nice question and very obvious question in current pandemic situation. But unfortunately, we have predicted some very important microRNA, but since we don't have a BSL-3 facility or we cannot work with SARS-CoV-2, so I did not test. But we have already predicted some very important candidates, and hopefully when I will have a facility, when I will have access to the facility, then probably I will work on that. Thank you.
Your work looks very promising, so it looks like we can always have that, working on this. So if I can ask you one question, which is by Dr. M.F. Khan. He asked that can a pathogen, a viral or bacteria, be a part of human microbiota? If yes, what can be the possible factors? So I good question.
Yeah. So it's a very tricky question. I can see that there is a use of the word pathogen. When we say pathogen, it is not friend. So yeah, if I will not use the word pathogen, then of course there are so many bacteria and viruses which can be associated mutually, and then they support each other. But when you say the word pathogen, then it is of course they are not good, they are not welcome, right? So yeah, but to elaborate more rather than going on words, so if we take some microbe and if they reach to the some place where they should not reach, then they cause the disease in some individual, right? Even normal microflora, if they will reach to the, say, peritoneal cavity, if there is a perforation in the gut, then that the microbe which is friendly, they turn to the enemy. So this depends on all the situation. It's not only about the word. It's all about the situation. So there is a restricted region, and they should stay over there. If they will reach to the some non-restricted region, then that leads to the complication.
I agree with you. So
I
you
think
can
I have answered adequately.
Put on your camera. Actually, you can share your put on your video camera so that people can look at you.
Okay, sure.
Okay, good. The other one is that what is the role of type II interferon?
So type II interferon is interferon gamma, right? So interferon gamma is basically produced by the activated macrophages, Th1 cell and NK cells, activated CD4, CD8 T cells. So they are playing much more important role in adaptive immunity, development of adaptive immunity. However, if the macrophage is treated with the interferon gamma or type II interferon, which is consist of interferon gamma member, then it will be activated. Its function will be enhanced manifold. So that what type II interferon.
So there is one more question, which says that any theory which governs the mutation in which region takes place and why. So you must have talked about some mutations. I don't know.
Yes.
The question can be, how are certain regions conserved in spite of mutations? So
Yes, this is a very good question, and I like to explain whosoever is very well. So
You will have to brief because we have to,
yeah.
just
So
concise.
I will quickly tell. So basically, what is happening when there is a there is an enzyme. Okay, so enzyme has a one active site, and there is a some surface amino acid. So if viruses cannot afford to make a changes in the active site of the enzyme. If they will make a change, then they cannot replicate. So they can only make a changes surrounding amino acid. Okay, in this case RNA polymerase PB1, and if it is and they can make a mutation in the surface protein like hemagglutinin, neuraminidase. Okay, but they cannot make a changes in the active site. So in a one of slide I have showed that this region is highly conserved where microRNA 485 is binding. Okay, so if they will make a change, then they cannot replicate. So that what all is.
Okay. So there is one more question on COVID-19. It says that it's not COVID-19 infection, is a misnomer. We should call SARS-CoV-2 infection. In SARS-CoV-2 infection, RIG1 overexpression is there or no?
So type 1 interferon is inducing the RIG. So there is some level of induction, although COVID has several proteins which inhibits the interferon induction. So if we mutate those proteins, then this will induce the. However, wild type will not induce so much RIG because they have a protein, some non-structural protein, which will inhibit the type 1 interferon production and subsequently the RIG expression.
So we have a question which says that what is the half-life of microRNA?
Yeah, this is a very good question. So probably you may or people are aware that this microRNA is now used for the diagnostic thing and so many other things. So in general, it is since this is a quite small and they are not present in free form. Please remember, they are present in bound form. So when the microRNA targets some gene, then there is a there is a complex which we call it as a risk complex, RNA-induced silencing complex. So generally, microRNA is not present in free form. It is present bound with the protein. So their half-life is quite long. It's not very short. It's not quickly disappear from the cell or from the system.
All right, Himanshu, I'll have to close here. Thank you very much for a very
Thank you very much for giving this opportunity. Thank you. Thank you.
Thank you very much.
Thank you.
And you can know. Thank you very much. So I move on to the next. You can close your camera and mic, please.
Yes.
Okay. So we move on to the next speaker, who is Dr. Arvind Sahu. Arvind is a senior scientist and PI at National Center for Cell Sciences in Pune. Dr. Arvind earned his PhD degree from the University of Delhi and did his postdoctoral training in the field of complement biology in the laboratories of Professor Michael Pangburn at University of Texas, Taylor, and Professor John Lambris at University of Pennsylvania, Philadelphia. Dr. Sahu is the recipient of Wellcome Trust Overseas Senior Fellowship in Biomedical Science in India and is a JC Bose National Fellowship. He very prestigious these fellowships he's had, and he was elected fellow of National Academy of Sciences Allahabad, Indian National Science Academy New Delhi, and Indian Academy of Science Bangalore. His research interests are primarily focuses on understanding virus complement interactions. His laboratory was one of the first to identify and characterize viral RCA proteins responsible for complement evasion. So today I've invited him to talk on basic complement system. So I wish he can explain that better and then come on end up with some of his research work. So Arvind, stage is yours for next 35 minutes. Arvind Sahu, please.
Yes, thank you, Sunil. Can you hear me?
Yes, we can hear you very loud. Loud and clear.
So let me share my slides. So Sunil, can you see my slides?
Yes, we can see that. Thank
Okay,
you.
okay. Thank you very much. So Sunil, for this giving opportunity to present. What I really love doing it is the complement system, which very few people really like to venture into this particular aspect of the immune system. So what I'm going to do today is, very in brief, give overall a sort of overview on the complement system. Is the what complement system is all about, what does it do, and what do we know so far about the complement system. But I'm not going to any specifics in particular, and rather would like to stick to the overview, and that is what I intend to do today. So what I'm going to cover today is in very brief, like in a slide, the early history. How did it all start? The complement activation pathways, then, all the three pathways in very brief. How the regulation occurs in the complement system, basically the self and the non-self discrimination in the complement system. A slide or two on the acquired immunity. How does complement, play a role in boosting the acquired immunity? And the complement evasion, how does pathogens evade the complement? And of course, in very brief, the role in the physiology, what complement really does, which very few laboratories really do and work in this aspect. So this all started in late 80s. So what people observed, a bunch of the labs and few researchers reported the bactericidal activity of a normal sera. What they essentially saw that if you take normal serum, normal human serum, and then incubate the bacteria with that, these serum have capacity to basically kill the bacteria. And this is the bactericidal activity of a normal human serum. But they could not, they did not really, could not find out why this activity is there until the Bordet, Jules Bordet, looked into that. And what he found that to have this particular activity, you need two components. One is the heat stable component, which is the antibody, and the another one is the heat label component, which is the complement. Okay, but he did not use the word complement. He used the word alexin. So initially, the complement was called alexin and not the complement. It was the Ehrlich who actually dubbed the system as the complement. So the complement name was derived afterwards, but the original name was alexin. And many people in the complement field still believe that alexin is a better name for the complement rather than the complement, but will not get into that. So this is when the whole thing started. Bordet not only showed that there is a heat labile component, but he also showed that this is a substance. He developed the complement fixation test and actually showed that the complement can be fixed on the pathogens. And so it is not just the activity, but it's the substance. But he actually thought it's a single kind of entity which is there, which later on we of course know that there are different pathways and more than 50 components are involved in this particular complement system. So how does a pathogen or a bacteria, or for that matter any surface, really activate the complement system? So let's say this is a bug circle, and then if you throw this in the serum, what will really happen? So if you take a bug and throw it in a serum, what will really happen is that complement can be activated by three different pathways. One is the classical pathway, the second is the lectin pathway, and third is the alternative pathway. And the job of all these three pathways, and we'll come to each one of them a little later, to make an enzyme called C3 convertase. What it really means is make an enzyme which can cleave C3, that is the central component. Okay. And what this enzyme does is it cleaves the C3 and then converts that C3 into C3A and C3B. And the C3B is the molecule that can go and deposit. When I say deposit, it is the covalent coupling of the C3B molecule to the pathogen surface by ester or the amide bond. So it's not just protein-protein interaction, it's actually the deposition is a covalent deposition, covalent coupling of the C3B molecule to the activating surface. And once this C3B is attached, then the complement system considers this particular pathogen, or for that matter, any surface, as a non-self, and then it activates, see, the AP alternative pathway kicks in, and then it can activate more C3. And what you have is a sort of a cover. The C3B molecule can cover and make a nice layer. And this is not just a mono layer, but C3B can deposit on the top of each other and can make really a thick surface. And that's how the complement can cover and activate pathway. And this can help in phagocytosis. This can help in elimination by the lysis and so on and so forth. So what really happens when the C3 is converted into C3B? For many years, we knew that there is a large conformational change. And this was done by using different monoclonal antibody, looking at the binding sites for various proteins. But we did not have really a good picture of that until the Pete Gross's lab saw the structure of C3B and showed that there is really a large conformational change. And if I show this movie, you will really appreciate what really happens. So what really happens is that the top part really actually moves to the bottom, and this model is called a puppeteer with a puppet. So what you really have is a body, you have a head on the top, and you have arm, and that arm is connected to the puppet, which is nothing but a domain called C3D. And this is the one which gets covalently, helps in covalent coupling of C3B molecule to the surface. So you can appreciate that there is a huge conformational change, which not only allows C3B to bind, but opens up a lot of the sites for the proteins to bind and activate the pathway. So let's look at the classical pathway. So for classical pathway to activate, you need what we all know, and you must have read it in the textbook, is the antigen antibody complex. And once you have antigen antibody complex, the C1 molecule, which is actually nothing but the C1q, C1r, and C1s, one molecule of C1q and two of R and S, they can bind to the antibody. But C1q or C1 molecule is not the only one that can actually activate the classical pathway, but you also have C-reactive proteins, SINR1, and sometimes C1q can even directly bind to the surface and can activate the pathway. And once this happens, the C1q is bound. What you have is the C1r, there is autoactivation that happens, and that clips the C1r, and that then activates the C1s. And once this happens, the activated C1s can cleave C4 molecule and can convert that into C4a and C4b. The C4b molecule can go on the surface. It again can covalently attach to the surface, and C4a molecule goes out. The C1s can also cleave the C2, and then it can convert into C2a and C2b. The C2a is the moiety that binds to C4b, and what you have is a C3 convertase. So this is how the classical pathway can generate the C3 convertase, which can cleave C3 into C3b. Now, for a long time, what you must have seen a picture in the notebook that what you have is, IgG, two molecules of IgG and C1q can bind, but now we have the salt structure, the cryoimmune structure of the IgG and C1q bound to that beautiful paper again from Pete Gross's lab. And what they showed that IgG actually forms a hexamer, right? So the C1q binds to multiple IgG. And then this is very important because if you mutate these sites, then the C1q cannot bind and the classical pathway activation cannot happen. Okay, so like what is there in the IgM, there is a multiple interaction of C1q with the IgG. The same thing also occurs when there is a IgG. And this is very important for the activation of classical pathway. Now, what about the lectin pathway? So now, as the name suggests, the lectin molecules are involved, and here the MBL or the ficolins or the CLK1, these are the ones which are the recognition molecules or initiation molecules in this particular pathway. And what obviously we all know that the lectins bind the sugar moieties, so they can bind. So like MBL, it can recognize the sugar moieties and also the ficolins, but they recognize a little different. We'll not get into that. So once the MBL is bound, it is associated with MASP. So like C1R and C1S, what you have here is the MASP, which they essentially do the same job, and they can, the MASP2 can cleave the C4 molecule, and then you can have C4b attached, and you can also have the C2a, which can be cleaved eventually by the MASP, and you can essentially get the same kind of converted C4b, C2a here, and that these can then, the converters can cleave C3 into C3b. Now, usually what has been seen is that in the MASP, they associated in the MBL, it is a homodimer of either MASP1 and MASP or the MASP2. So the question then, how does the MASP2 is activated? The original idea was that MASP2 is auto-activated, but then, the paper came out, a beautiful paper in PNS showing that there is this juxtaposition activation. What it means is that the MASP1 cleaves a neighboring MASP2 and then it activates that, and then that's how the MASP2 works. What about the alternative pathway? So this is very interesting pathway in the complement field, or for that matter in the physiology, whereas there is no requirement of any initiation molecule or activation molecule. So what really happens that the C3 molecule has an internal thioester bond. And when this internal thioester bond reacts with the water, then it gets hydrolyzed. So this is protected normally in C3, but the molecules breathe, and every once in a while the water molecule can get inside and can cleave this, activate this thioester bond and converts that. And when this happens, there is a large conformational change, which is essentially like C3b. So the C3 water molecule, what we call, or C3b like C3 molecule, which is generated, is essentially like C3b molecule, but this is in the fluid phase. So when this happens and you get C3 water molecule, then it can basically bind to factor B, which is like C2, and then this B can be cleaved by another protease called factor D, and what you essentially get a C3 convertase, but that is in the fluid phase, C3b water BB. And this C3 convertase can act and cleave C3, and as this C3 is cleaved, the C3b generated can then bind to the surface and can generate again the C3 convertase by binding to factor B, and properdin can stabilize this one. And once this happens, then the C3 convertase can cleave C3 and C3b is deposited. So basically for the AP, it differs in that it doesn't require any initiating molecule. And once you have a surface that is pretty good in binding to C3b, then the C3 activation can happen or complement activation can happen. Okay. So what about the further pathway? What we have is that once C3 convertase is formed, the additional C3b molecule, when it attaches, it converts into C5 convertase, which can cleave C5 into C5a and C5b. The C5b generated can bind to, it has a labile site, which can bind to C6 molecule. This dimer can then bind to C7 molecule and can get attached to the surface, which you can see here. And then once it is attached, the C8 molecule can bind, but it can penetrate the membrane. So it becomes a little leaky. And then the C9 molecule comes, and then there is a polymerization of C9 leading to a pore, which is about 100 to 110 angstrom. And then, of course, there is a lysis. This structure has also been solved in 2015 from, of course, 2 different labs. I'm just showing one set of data. So what initially people thought that when you have a pore, it's primarily formed by C9, but that's not true. Really, the pore formation, C6, C7, C8 are also part of the pore. And this is a split washer kind of arrangement, which is there about 110 angstrom, roughly about 18 molecules here, which you get, which participate, in the formation of pore. So here, all these are C9 molecules, which you see it in blue, and that is resulting in a pore formation and the lysis. So the question obviously is that if the C3b can bind to a surface, when there is activation is happening, then how does a complement really discriminates between the self and non-self? So initial there were 2 sort of thoughts. One is that when C3b is attaching to any surface, we must have sugar moieties or hydroxyl groups or the amino groups which the C3b prefers, right? And this must differ. The sugar moieties must differ, or the amino groups which are there, the molecules, the orientation or something has to be different in the healthy cells and in the foreign cells. And therefore, when the C3b attachment happens, it can actually differentiate what is self and non-self. And the other thought was that no, that's probably not true, and it could be just the regulators on the surface. So the healthy surface must have certain regulators which regulate. So both of these things were tested. I was actually, in particular during my postdoc, tested this hypothesis about the C3b attachment, and what I found is that okay, there can be difference in the C3b attachment, but ultimately the C3b attachment can happen on even healthy human cells pretty well, and therefore this is not the mechanism how the non-self really, self non-self discrimination happens. So C3b can well attach to the human healthy cells, but regulators are there. The cells are lined with the regulators, and then they can inactivate the C3b very efficiently, and that's how the complement system is not activated on the healthy human cell, but can happen on the foreign cells, or even when the cell is undergoing, let's say, apoptosis. So there must be then proper regulation in place. And is there proper regulation? Then the answer is true. The regulation, which is main enzyme convertase, a lot of regulators are there which basically regulate the C3 convertase. You also have a C1 inhibitor which can act as an initial and can act on the C1R and C1S enzyme, basically stop the activation, can also act on the MASP. We also have regulation at the C3 and C5A level, basically carboxypeptidase N, which can remove the terminal arginine and convert into desage forms, and therefore there is no active, these molecules become inactive or very, very less active. And then also the CD59, which can basically bind to C5b through 8 and then stop the C9 polymerization binding and the polymerization. So the regulations are pretty much there in the place, and the main mechanism of regulation is at the C3 convertase level, and this family is called the RCA, or regulators of complement activation. What they do is that basically when the C3 convertase is there, which is bimolecular, then these molecules bind and decay it, and this is called decay activity. And another way they can act is act to the one of the subunits, which is C3b or C4b, and can recruit another protease called factor I, and basically which can clip and inactivate and convert it into inactive form of the C3b or the C4b. So we looked into this using the viral molecules, is how this process really happens in our lab. And what we saw, of course, a lot of different labs have contributed onto different aspects. I'm just showing our data and talking about the molecular events that are involved for the cofactor activity. So what these molecules basically do is, these regulators, they bind to C3b and they actually bridge So what we have shown that bridging is very, very important. So this is sort of a body, and this is sort of an arm of the C3 molecule, and these molecules bridge. And this is very important because the molecule is very flexible. And once this happens, then it can recruit a protease, which is a factor I, again interact with the factor I. And when that happens, it can clip here in the arm, okay? And that's where the factor I can cleave. So two steps are important. One is the bridging, and second is the recruitment of factor I. So these regulators basically do these two jobs. Now, what about these two activities? I'll not go into decay activity, which we have also contributed a bit. But what we wanted to, I want to show that, when you have viral molecules have only 4 and they have both this activity, and what we have shown that there's a functional modularity. So you have initial domains, let's say 1 and 2, which are very important for the decay activity, and 2, 3, which are very important for the cofactor activity. And if you, same thing is also true for the human molecules, although we have shown this with the viral molecules. But if you take human molecule and take these domains out and put it together, you can generate a very effective and very active regulator, which can have both the activity, these which I have shown, and you can generate by putting these domains together. But I'll not get into details into the mechanism of this. So if the regulators are so important, then what happens if the regulators are not there? In other words, if there is a polymorphism, or deficiency of these regulators, then do we have a disease? And the answer is yes. You have, this is like age-related macular degeneration, AMD. In the eye, you have degeneration there. You have C3 glomerulopathy. In the kidney, you also have a typical hemolytic uremic syndrome where factor H is mutated and is not functioning, or where the autoantibodies are generated against factor H. And we have a lot of these cases. In Professor Arvind Bagga at AIMS is looking into these and shown that as many as 50% of the kids who have these AHUS have the autoantibodies against factor H, and have all sorts of problems because of that. So the complement system, how does it work? The complement system not only works as innate immunity, that means it is involved in opsonization, in the lysis or chemotaxis because of C3a, C5a, generating inflammation, but it can have a lot of cellular activation, right? So it can have chemotactic activity against the neutrophils and monocytes. So for example, C5a or C3a can have against mast cells, can interact with so many different kinds of cells, and also activate them. Also, not only that, but the APC like, macrophages, DC can also act on B cells. And now we know that, in last 10, 15 years, a lot of work has been done on how complement can interact or bridge innate and acquired immunity. And I'll show a slide or two on that. So what has been shown, it's a long time back, it was observed in 70s that if complement is not there, or if you have a C3 depleted, animals, or if there is a C3 deficiency, the antibody response is very low. But then it took many more years, for Doug Fearon's lab to show that basically, if you have antigen coupled to C3d or complement fragment, then what you have is a bridging between BCR and CR2 on the B cells, and that reduces the threshold of B cell activation, and basically you get more. So if you, in other words, what they have shown beautifully, that if you take hen egg lysozyme as an antigen and couple C3d, then just by coupling, 3 C3d molecules, you can reduce the antigen, 1,000-fold, and you can have, if you don't have, you just take antigen per se or antigen with the C3d, there can be 1,000-fold more generation of the antibody response in vivo. Later on, it was also shown that the CR2 is also then on the FDCs, and that can help in antigen retention, and of course that can help in the germinal center and B cell activation and more antibody production. Also, when there is a T cell and the APC interaction here in, for example, DC, when this happens, the complement synthesis is there, And both these T cells as well as DC have C3A and C5A anaphylatoxin receptors. So when the activation happens, when this DC T cell activation they are interacting with each other, C3 and C5A can act on these. And when that happens, for example, if complement is there, you have basically mTOR activation leading to less of TGF beta, and what you have is activation of the T cell depending on cytokine melu Th1 or Th17. And but when the complement is absent, then instead of that, what you have is more TGF beta and more of a Treg and not Th1 and Th17. So what it has been shown is that uh, complement is not only important for B cell, but also T cell and polarization of T cells. If you don't have complement, and I like this catch line is the complement a day, keep FOXP3 away. Very recently, again, in the last few years, so all these years, what we know, the complement is activated outside the cell. What has now been shown by Claudia Kemper's group that complement, of course, is synthesized by most immune cells and also non-immune cells. But when complement is getting activated inside the cell, in this particular case, we are talking about Th1 cells. So let's say when the cognate recognition is occurring and also CD46 ligand is there, and that is also you have a bridging of that along with the TCR, which is happening. You have C5 activation inside the cell, and the C5a R1 receptor, which is inside the cell, and we don't really know exactly which compartment we are talking about here. But what we know that the C5 is released and it is interact with the intracellular receptor. You have a ROS and you have NLRP3 inflammasome activation. And this is obviously you have the IL-1 beta there and then the Th1, that is IF and gamma induction. And when you have R2, for example, on the surface, C5a R2 receptor, then the C5a can go and that can lead to the contraction of Th1 cell. Beautiful paper came in Science. What I'm trying to emphasize here is the complement is not only important when it is activated outside the cell, but it seems that complement is also important when it is activated inside the cell, and that may have something to do with Th1 cell generation and T cell activation. Now, here is one example from our lab, what we wanted to really look into that. Can we really demonstrate, because our interest is the virus, take an acute virus, flu, for example, take a virus where we get infected and in, let's say, 7 to 10 days, we are fine. What it means is that our body is capable of taking some these kind of viruses, and therefore they don't really have great immune evasion mechanisms. And flu, of course, we know that it doesn't have any immune evasion mechanism as far as complement is concerned. So in this particular, this is just one slide I'm going to show you. So what we did is that we did this is the pandemic influenza infection in the mice, either the normal or the complement deficient mice. If you see the C3, which is the central component, if you do that, normal mice, you see that, you have a weight loss and they recover fine. There is absolute no mortality if you look at the panel below. But if you see the C3 knockout mice, they start dying, and pretty much in about day 10 or 11, all of these mice die. They don't survive, right? And if you see just the component C4, which is a component of the lectin or the classical pathway, you also have a weight loss and death, but not 100% death. And if you take a factor B, there also you see death, but not 100% death, about 20 to 30% death. And so is a meaning that if one pathway is absent, the other pathway can take over and can protect the mice. But if you have C3, which is a central component absent, which is almost like complement knockout, then you see that all the mice die. Then the question is why they die. What we have shown is that uh, complement is neutralizing this virus directly with the help of classical pathway, and also the activation of AP, which is taking over but you do need the antibodies here. What we have also shown is to generate these antibodies, you need complement. So we have shown that if you take, for example, look into the C3 knockout mice here, you don't really get the specific antibody generation against the HA, which primarily in this particular case are IgM type, and you don't have that. But if you actually take from the wild type and put it into these mice, you can rescue. So these antibodies which are generated, so complement is helping in generation of the HS specific antibodies which can help. We did this work in NCCS with the help of Dr. Girdhari Lal, and somehow I'm, trying to see if other people also get excited into the complement field like me, and at least I could perceive Girdhari to look into this aspect. So thank you, Girdhari, for that. Now this is if this is true and pathogens, evade, they obviously have evasion mechanisms, and certainly the viruses, which have also, they mutate a lot and they can evade a lot, and they have sort of evasion mechanisms for almost all the immune pathways. What about the complement then? Do they really have? And yes, the pathogens do have, for example, staph can deplete IgG, or bugs can, or bacteria can have different molecules which can target different complement molecules. For example, here, C3, C5, in staph or in the streptococcus. And viruses are also known to recruit the complement regulators from the host cells, and they make their own cells. And our lab particularly is interested in this, and my lab is focusing particularly on this aspect of both the pox viruses and herpes viruses have these mechanisms. So these are particularly, smallpox virus or the variola virus, and of course the vaccinia virus also has this. And of course, bugs can also have enzymes which can degrade the complement protein. So various different mechanisms pathogens can develop and can evade the thing. Now a little bit about the role of complement in physiology in general. So this is one particular model which we tested here, and of course other laboratories have also mentioned that, for if you take the osteoclast precursors, they make fuse polykaryons and they activate osteoclasts. So in other words, what we know for many years now and decades now, that the myeloid cells can synthesize complement component very well. So is there any role of complement in polarization of these cells is what we were trying to ask. And in this particular osteoclast, so in the formation of osteoclast, whether the complement has any role. And what we saw that if you take C3 knockout mice, very few osteoclasts are formed in vitro when you incubate with the MCSF and rankle. And you can see here on the right hand panel, the giant osteoclasts in wild type, but very smaller ones in the C3 knockout. But if you reconstitute with the C3 and C5A, you can actually regain this. So basically, C3 and C5A help. I'm not going to go in detail of this, but this is one. It has also been shown that in the brain development, the complement, particularly C5A, is playing a role. So, if you have deficient mice, if you take, for example, folate deficiency, we know that can have a neural defect or neural tube defect. So in this mice, if you also take out the C5A, you know, either you use the receptor antagonist or you use the deficient mice, which are C5AR1 deficient, receptor deficient, the malformations are increased significantly. And here you can see some of those on the right panel, where you can see that folate deficiency plus the C5A receptor deficiency can really, exacerbate this particular effect. This is one example. The last one I'm going to show you here is that it can also play, role in chemotaxis here, particularly the C3A. Beautiful paper from, developmental cell can act as a chemoattractant for neural crest cells, which happens during development. So here, let me see if I can play this. Sorry. So what you can see here is that if you have C3A, these cells basically aggregate and move beautifully in C3A. And if you take the Desarge version, really then the cells don't go there. So even in the development, see, complement seems to have a play a role, but I would like to point out here is that there is a redundancy built in our system. In other words, that if you take complement knockout mice, their development is not really getting affected. The phenotype is not there, but then there is a redundancy obviously there, like you saw in folate. So if you don't have folate and complement, then only you see this particular phenotype. If you just have complement deficiency, you don't see the phenotype. So one needs to keep that in the mind. Thank you very much. I did not show too much of my data. I just, because it was for students, I thought it would be better if I restrict to just basics of complement and try to put everything together what complement does. So thank you very much, and I would like to stop here.
Thank you, Arvind. Nice talk because complement, as you said, makes a very important component of innate immune response. But not only that, it's a bridge between innate and acquired immune response, adaptive immune response as well. Because if there is no complement, you have shown that uh, the diseases cannot be handled very nicely. And with the mouse model and with the knockout mice, you have shown that it's so important to have these complement components that it needs activation to generate a specific immune response. So it was very good. I'll need to ask a few questions. If you can take off your slides and join us with the camera. Thank you.
Okay.
The question comes from Dr. Krish Ghosh. He wants to know that he always ponder over the term takeover.
Yes.
Exactly, C5 and C5A to C5B. So can
Okay.
you?
Sure. So I thank you for pointing that out, because that term is very often used. What takeover basically means is that the hydrolysis of C3 molecule with the C3 water. So what we have is C3, a very high concentration of C3, about 1.2 milligram per ml in our serum. Very, very high, one of the highest, proteins. So proteomics people don't like, actually. They want to get rid of all the complement proteins before they analyze anything. So what happens that, as you know very well, that the water is very high concentration, 55 molar. Okay. So what happens that the water can, once in a while get inside the thioester pocket and can then break the thioester bond. Okay. And when this happens, we call it as a hydrolysis, or also other name for this is the takeover process. And when this happens, the conformation of the C3 changes and it becomes more like C3b, and therefore the alternative pathway convertase can be formed into the fluid phase. So basically, the takeover essentially means the hydrolysis of C3 or conversion of C3, native C3 molecule, into more of a C3b like a molecule. I hope that is clear.
Okay, yes. Second question is again similar because this was by another person who wants to ask the water molecule playing role in activation of alternative pathway. So you've already explained that the water molecule does play a role, and how it does is a takeover process. So hydrolysis is done. So then there is another. Can you open the question answer box? You can open it, and there are a couple of more.
Let me see if I can do that from here.
Bottom, of your screen, question answer box.
Question answer box. Yeah.
Q&A.
Okay. So where are we right now? Takeover we have answered. Water molecule we have answered. Can I have a link of video or the paper and the author model moving covalent coupling? Yes, so this is a Pete Gross's paper. You just send me a mail and I'll send you this. So Sunil, please share my email ID so I can do that. Or I'll just tell Arvind Sahu, ARVIND SAHU at nccs.res.in. Please send me a mail and I'll share all the links. Okay. Is there any role of anaphylatoxin which support complement system? So anaphylatoxins basically, once they are generated, then their job is to bind to the receptors which are present on immune as well as non-immune cells. Okay, very important. I did not cover that aspect, so we all know that that means, for example, neutrophils are getting activated, macrophages are getting activated. There is also chemotactic activated. They are there. They enhance all the activity, all sorts. The cytokines which are there for after the activation, inflammatory cytokines in particular. When you have an apoptoxin generation, you have all these more. But there is also something that happens. For example, anaphylatoxin, both C3 and C5 play a very important role in liver regeneration, right? So if you don't have complement, you don't get really very good liver regeneration. So it also, these anaphylatoxins, but directly in terms of complement activation per se, no. But their role is secondary. Their role is to interact with the immune as well as non-immune cells and activate. And sometimes this can cause problems. You can have a lot of inflammatory cytokines generated, and can have a problem. And one such problem, of course, has been seen in COVID now. You have a lot of innate system activated, certainly complement system getting activated. Some papers have been published, some are in the pipeline. You can look into that. And what has been, so basically people believe, well, I should not say that. Some privileged information is there. I will not tell you about that, but complement is getting activated. People have looked into this by using in humans the regulators. C5 monoclonal antibody has been tried. There is a molecule called comstatin, which I happen to find it out or discover in John Lambris's lab, which is done wonders in phase 3. Both have been tried actually in humans, and those have shown promise. Okay, so yes, innate immunity, DC3 complement and the toxin can help and can also damage. Needed reference for covalent coupling of C3b. A lot of references there. Originally, Brian Tack discovered the original paper came in JBC, but there are many papers on this. Please send me a mail and I'll share all the references you want. And a lot of work has been done by Law and Levine. The interreaction mechanism came into nature by Law. I will share that too, as how the ester or the amide bonds are formed. I didn't get into detail of that. Tick over, I have already answered. What is the terminal complement deficiency? How is complement deficiency treated? Okay, so terminal complement deficiency, basically complement and particularly terminal, what you have is a bacterial problem, infection, Neisseria infection in particular, right? So plasma replacement was something which was tried, and has been helped in the past, and that is what has been done. But then you have to do that repeated. But basically, when you have complement deficiencies, what you get is more Neisseria infection, but not certainly more viral infection, right? Why? Because two reasons. People did not look into that. I have shown one example to you of flu, the pandemic H1N1, where the complement deficiency can, and another, a lot of these have evasion mechanisms. So it doesn't matter to many viruses whether complement is there or not. Is there any polymorphism in C3b and its Fc binding domain? Polymorphism C3b certainly are there. Many examples, I'll not get into that. And there is also blood group differences because of those. I will not get into that, but please send me this. Fc receptor in particular, no, I have not come across any particularly, but polymorphism which is affecting Fc binding. Is there liver damage, how complement mediated immunity is affected? I have answered that complement is important in liver regeneration. A lot of work. This was something which came by surprise in John's lab. And I suggest students, do these interesting experiments. I don't know if I have time to narrate the story, but I'll tell you, one girl moved from Greece, to John's lab, and she had a friend who used to do the liver regeneration. He was a very good surgeon. What they used to do is to cut the liver, take the 70% of the liver off, and then took, and their lab discovered that IL-6 plays a very important role. And this is the time when we had got C3 knockout from Harvard, from Mike Carroll's lab. And what she did is that we had she asked me, can you spare a couple of mice to me, Arvind? I said, yeah. She didn't tell me what she wants to do. And with the help of this person, she chopped the liver, and for sure, all the 3 mice she did, of them died. And then later it became a huge program in John's lab to look into the liver regeneration and this. So yes, complement plays a very, very important role in liver regeneration. Can complement activation be used in immune therapy against CoV, SARS-CoV, specifically 2? Yes, I already mentioned people are thinking, trials are going on. A bunch of cases have already been tried and reported. One particular is monoclonal in antibody against C5, and again second one Comstatin, which has been tried with good, but these are only few patients, 2 patients and 1 patient. You can't really say anything. All you can say that it helped this particular patient, but there has to be a systematic study to really evaluate the role. So as of now, the trials are going on, but we'll have to wait and watch. So I guess I answered all the questions.
Yes, thank you very much. It was very nice talk and very elaborate, and you handled the questions very nicely. So most of things are covered. Thank you very much for giving such an elusive talk, and I'm sure all the participants have enjoyed this. This is very important. So thank you very much. We need to close the session now. And before I close, thank you, Arvind. You can
okay.
log.
Yeah, thank you, Sunil. Thank you very much for inviting me to talk about complement. Not many people really want to cover complement, so thank you very much for that.
You are one of very rare people who work on complement, and so that's why I always like to invite you on this.
Okay.
Thank you.
Thanks. Bye.
So, okay, so now we have a lunch break for about 1 hour, and we'll assemble back again in 1 hour. So we all can have lunch, and we'll get back to you. Thank you very much. We see you again.
Good afternoon, everyone. We welcome you back after the lunch session. So we start with the session 3. And this session will be moderated by Dr. Amit Awasthi. Dr. Awasthi is Associate Professor in Immunobiology Laboratory, Translational Health Science and Technology Institute, Faridabad, India. Dr. Awasthi did his postdoctoral training at Harvard Medical School in USA. He is the Secretary of Indian Immunology Society and Council Member for FIMSA. Dr. Awasthi's research interests are understanding T-cell differentiation pathways in immunity and autoimmunity and cancer immunity, and understanding the metabolic regulation of T-cells during their differentiation. So I now hand over the stage to Dr. Awasthi to take over the program and moderate the session 3. Thank you, Dr. Awasthi.
Good afternoon, everyone. Thank you, Gul, for a kind introduction. And I guess most of us, the participant, I can see around 155 at this point of time. So people are trying to come back. So this session, which is a session for basics of immune system, this is a session 3, and we have 2 great speakers. The one, Osamu Takeuchi from Japan, and second talk followed by the first talk by Professor Mani Al Gupta Sarma from PGI Chandigarh. So I would just take an opportunity to introduce Professor Takeuchi, who is actually at Graduate School of Medicine, Kyoto University. So basically, Dr. Takeuchi, I hope I'm pronouncing it correctly. Pardon me if it is not in a Japanese way. Is a professor.
No, you're perfect.
Professor in Kyoto University. He has received his MD from Osaka University. And clinical training, he entered Graduate School of Medicine, Osaka University, where he started working on innate immunity with Professor Akira, and where he actually worked a lot on toll-like receptors and how these toll-like receptor can actually senses the microbial component. And he received a PhD degree. Subsequently, he moved to Boston, where he did his postdoctoral training at Dana-Farber Cancer Institute. And after that, he became the assistant professor in Osaka University, where he worked on functional role of TLR signaling molecules and Rig-like receptors in inflammation. In 2012, he moved to Institute for, it's called Frontier Life and Medical Sciences, Kyoto University, as a full professor. He's currently focusing on the post-transcriptional regulation of inflammation by a set of RNA binding protein. Uh, Professor Takeuchi has received a prestigious Japan Society for Promotion Prize and Japan Academy Medal on 2016. He has been one of the very highly cited researcher according to the Thomson writers in from 2014 to 2017. So basically, it's kind of, I think he has professed the area which he's going to talk about, and he will be today talking about the regulatory mechanism of innate immune responses. So, Professor Takeuchi, I welcome you for this, and please take over this.
Hello.
Yes, we can hear
Can I
you.
share the slide? Now is it okay? Can you see my slides?
Yes.
Okay. And thank you very much, Amit, for your kind introduction. And also thank you for this kind invitation to this great FIMSA immunology course. And thanking Sneo. And my name is Osamu Takeuchi from Kyoto University. And the title of my talk is The Regulatory Mechanisms of Innate Immune Responses, as he introduced. And so in the morning sessions, I think you guys have a nice introduction about how innate immunity works. So probably I don't need so much of the introduction, but here I will start from how macrophages sense the pathogen infection. So these innate immune cells, such as macrophages and dendritic cells, sense pathogens through a set of pattern recognition receptors, like toll-like receptor, and also some cytoplasmic sensors of viruses or bacteria. And once they sense the invasion of these pathogens, they initiate the intracellular signaling pathways leading to the transactivation of the messenger RNAs encoding the cytokines such as TNF, IL-1 beta, IL-6, Type I interferons, and so on. And these develops the inflammation. And, the inflammatory responses are, of course, really critical for the clearance of invading pathogens. But on the other hand, if the excess and chronic inflammation occurs, so this is going to the development of the pathologies like septic shock, autoimmunity, and metabolic diseases. So therefore, at least in the physiological conditions, these inflammatory responses or the innate immune responses are tightly regulated. And in terms of the pattern recognition receptors, so in addition to the prototype toll-like receptors, which are present on the cell membranes and sensing various pathogenic components from bacterial lipoproteins, lipopolysaccharides, flagellins through a set of toll-like receptors from 1 to 5 and 6. And then a set of TRLs present on the endosomal membranes like TRL 3, 7, and 9 senses viral double-stranded RNA, single-stranded RNAs, and CpG DNA respectively. And in addition to these TRLs, there are another classes of the pathogen sensors, pattern recognition receptors are present, like RIG-I-like receptors, comprised of RIG-I, MDA5, and LGP2, and sensing the viral long double-stranded RNAs, and or the 5' triphosphate and double-stranded RNAs. And in addition, there's another set of the pattern recognition receptors, like node-like receptors are present, and they are also subdivided into node 1, node 2, or the NLRC4 or NLRP3 type node-like receptors. And there are many other NLRs. And in the case of node-like receptor 1 and 2, so they sense the component of the bacterial peptidoglycan to induce the cytokine gene expression. But on the other hand, another classes of NLRs like NLRC4 and NLRP3 are recognizing either fragilin or the crystals from even dumps and activating the inflammasome that is going to the maturation of island beta. And even more, there are another classes of the PLRs, like C-type lectin receptors are present on the cell membrane and comprised of the dectin or minco receptors, sensing the beta-glucan or TDM from Mycobacterium. And finally, even there's a cytoplasmic double-stranded DNA sensors from DNA viruses are present, which is a famous C-gas protein. And most of these pathogen sensors are going to the activation of intracellular signaling pathways. And of course, this is an example of the TRL signaling pathway and really a simple scheme. But anyway, the TRL signaling is leading to the activation of transcription factors, especially NF-kappa B, through a kinase called I-kappa B kinases. And then this NF-kappa B transactivate the cytokine messenger RNAs, translated into the proteins and are secreted to the out of the cells. But if the too much cytokines are really harmful to the cells, this pathway is really tightly regulated by multiple mechanisms. So several of these mechanisms are the inhibition of the signaling pathways and also epigenetic regulations. And this is a bit more detailed scheme of the TRL signaling. And following the TRL activation, MID88, which is an adapter protein of TRLs, recruited to TRL toll-like receptors, and then activate kinases called IRAC1, 2, and 4, which also subsequently activate TRAF6, the ubiquitin ligase, and followed by the activation of another kinase called TAC1, and leading to the activation of the I-kappa B kinase complexes. And indeed, when this signal is activated, a set of the proteins like SOX1, ILAC-M, and TANG-A20, these proteins are induced in addition to the cytokines. And then these proteins are known to interact these proteins. For example, in terms of SOX1 and IRAC-M, they can interact with IRAC1 and 2 and inhibit its kinase activation. And in the case of TANG or A20 sealed, they are suppressing the ubiquitin ligase activity of TRAF6 to inhibit its signaling pathways to cease the inflammation. And in the nucleus, there are another set of the regulatory mechanisms, especially in epigenetic regulations. In terms of the histones, so histone modification like maturation of the histone H3 K4 trimaturation or histone H3 K419 acetylation known to be the related with the active transcription. And on the other hand, the H3K27 trimaturation is related to the suppression of the gene expression. And multiple proteins and also long coding RNAs, a set of the non-coding RNAs are involved in the regulation of this gene expression at the histone modification levels. And not mostly, even the set of the proteins initially induced by the TRL stimulation, like ATF3 or JMJD3, are known to regulate regulates the this histone modification as a negative feedback pathway. In the case of ATF3, the which interact with histone deacetylase HDAC1 to inhibit the histone acetylation, to inhibit the cytokine gene expression subsequently. And in the case of JMJD3, which is the methylase for the H3K27, is on the other hand important for the control of the macrophages, especially the differentiation of the M2 type macrophages involved in the allergic inflammation. And in addition to this type of mechanism to regulate the gene transcription of the cytokines, still the messenger RNAs encoding cytokines are also tightly regulated by the messenger RNA decay. And I'm going to focus on this pathway mostly in this talk. So I'd like to emphasize the importance of this mRNA decay in the control of the cytokines in macrophages. So this is a bit busy data, but showing the half-lives of the messenger RNAs in macrophages. So this is messenger RNA half-lives of resting macrophages. So of note, the messenger RNAs of housekeeping genes like beta-actin or ribosomal proteins, so they are really stable and not degraded. But on the other hand, a set of genes encoding cytokines like TNF, alpha, beta, or even IL-6 messenger RNA, so their messenger RNA half-lives are really short, and sometimes it's about 10 minutes or 40 minutes. So really rapidly degraded. And also the other interesting thing is following the activation of macrophages with LPS, the TLR4 ligands, so the half-lives changes and are mostly extended to facilitate the production of the cytokines when needed. So the question is, what kind of mechanism is governing this mRNA decay in macrophages? So this is a general cartoon of the messenger RNAs. And messenger RNAs are comprised not only of the coding sequences, but also 5' and 3' untranslated regions, UTR. And in terms of the cytokine or inflammatory messenger RNAs, it is known that there are some specific sequences like AUUUA sequence called AU-rich element, or some specific stem loop structures that can be recognized by a set of specific proteins like TTP and AUF1 for the AU-rich element. And in the case of some stem loop, the protein called ROQUIN recognizes it. And furthermore, it is well known that the microRNA binding sites are enriched in the 3' UTR of the cytokine or inflammatory messenger RNAs. So when these regulators are recruited to the mRNAs, they what they do is they recruit the RNases to the messenger RNAs. So one famous messenger RNA degradation complex is CCR4 node, which function as a deadenylase to remove the poly-A tail from the messenger RNA, or also include the decapping enzyme to remove the cap structure on the cytokine messenger RNAs. So when these messenger RNAs that lack the poly-A tail, then they are further degraded by the exosome, which is degrades the mRNA by the exonuclease mediated manner. So the importance of this system is characterized by the cause of the severe autoimmunity in mice lacking TTP or ROCK. However, we are focusing more on totally independent system, but mRNA regulatory systems, which is mediated by a protein named regnase 1. So this regnase 1 is functioning as a endonuclease to cut the mRNA in the middle and degrade the messenger RNA independent of the CCR4 node complex. So what is regnase 1? This protein is encoded in a gene called ZC3H12A. And in addition to the CCCH zinc finger domain, which is known to binding with RNA, it can it harbors a nuclease domain whose structure is like this, forming a catalytic site with 4 aspartic acid. And indeed, this protein has an activity to degrade even the circular RNA, which means this is not the exo, but the endonuclease. And also a 1-point mutation of this aspartic acid in a catalytic site leads to the abrogation of this RNase activity. So indeed function as RNase through this domain. So what is the function of this RNase? So to make a long story short, we found that this regnase 1 function as a RNase to degrade a set of cytokines like IL-6. So as you can see, the regnase 1 overexpression leads to the rapid degradation of IL-6 messenger RNA in a manner dependent on its RNase activity. So when we deleted the regnase 1 in mice, the macrophages from the regnase 1 knockout mice induced high amount of the IL-6 in response to LPS stimulation. So indeed, even in vivo, the regnase 1 is important for the control of IL-6. And more interestingly, so owing to the hyperactivation of the immune system, so the regnase 1 knockout mice spontaneously develop severe autoimmunity characterized by the infiltration of the immune cells into various organs like lung, and also a production of the autoantibodies like the anti-nuclear antibodies. So these results indicate that the regnase 1 acts as a novel brake or negative regulator of the inflammatory responses in macrophages, which degrade the cytokine messenger RNAs like IL-6, IL-12B, and in addition, the PTGX2, which encodes COX2, or NF-kappa B-IZ, encoding I-kappa B-zeta, regulator of the inflammation in macrophages. So they are all degraded by the regnase 1. And in the absence of regnase 1, the stabilization of this messenger RNA leads to the hyperproduction of the cytokines. But story was found to be a bit more complex because in the regnase 1 is also expressed in the acquired immune cells like T cells, and in T cells, regnase 1 degrades a different set of the genes like interleukin 2 and cell surface activation co-stimulatory molecules like ICOS, OX40, and even the transcription factor serial messenger RNA can be degraded by regnase 1. And in the absence of regulation in T cells, it can also lead to the hyperactivation of the T cells and also contributing to the development of the autoimmune diseases. So another question of this type of the post-transcriptional regulation is how these post-transcription regulators select the target mRNA for degradation. And we discovered that this regnase 1 is also binding with a set of the stem loop sequences present in these cytokine or inflammatory gene mRNAs. And and also we also explored the mechanism of the regnase mediated mRNA degradation, and found that the regnase 1 degrade rather translationally active messenger RNAs in the ER, the endoplasmic reticulum, and binding with the ribosome. But on the other hand, so this stem loop is not only act as a recognition sequence for regnase 1, but the same sequence was commonly recognized by another RNA binding protein, ROCK1. But their localization is totally different. In the case of ROCK1, it is localized in a stress granule or P-body, which function as rather a storage of the mRNA, and leads to the degradation of messenger RNA in a translationally inactive manner, inactive mRNAs. So this means that so in macrophages or immune cells, so there are multiple breaks even in terms of the messenger RNA decays. And it looks that regnase 1 is stopping the actively translating messenger RNAs, but on the other hand, ROCK1 seems to act like a handbrake for the mRNA decay and the inhibition of the inflammatory responses. And another interesting thing is that this regnase 1 is acting not only as a constitutive mRNA degradation machineries. So indeed, in resting cells, the regnase 1 act as a negative regulator. But when TLRs are activated or the IL-1 or IL-17 signaling is activated, which commonly activate ICAC kinases, it can directly phosphorylate the regnase 1 through the conserved DSGXXS sequence. And this phosphorylation is leading to the ubiquitination of the regnase 1, followed by the degradation by the proteasome systems, facilitating the robust production of IL-6 when needed. And even in T cells, the regnase 1 is tightly regulated, but in different mechanisms. In T cells, the MOT1 protease can degrade or cleave the regnase 1 to inactivate to facilitate the T cell activation when TCR recognizes antigens. So, what is the function of this regnase 1 in human diseases? So we recently discovered that the regnase 1 is involved, regnase mutation is involved in the pathology of the ulcerative colitis and inflammatory bowel disease, which is causing the persistent inflammation in large intestine, causing the severe destruction of mucosa and the formation of the ulcers. So because of this inflammation, so there's a damage or deficit of the crypt happens. So crypt is a unit of the differentiated epithelial cells, the intestinal epithelial cells, together with stem cells. So when this crypt is missing, so then that was repaired by the fission of the surrounding these crypts. So the hypothesis was in these UC patients, the dynamic remodeling the crypt happens. So this could be this recurrent proliferation is leading to the accumulation of any beneficial mutations. So therefore, we collaboration with Dr. Seiji Ogawa's group performed whole exome sequencing of the UC patients compared with the control and checked if any mutations are accumulated. So indeed, in the UC patients, so there's a huge accumulation of multiple genes. And interestingly, many of these genes are involved in the IL-17A or F signaling, like IL-17 receptors or NF-kappa BIZ, which is important for IL-17 mediated inflammation. And in addition, we found a mutation in the Regnase-1, the GC3812A. And interestingly, the mutation hotspot was found in this DSGXXS motif of the Regnase-1, which is a phosphosite by the kappa B kinases. So what is the function of this for the mutation? So indeed, this mutation is leading to the degradation of the Regnase-1. So the mutation might inhibit the degradation, which was confirmed by the experiment of the expression of mutants. And we also found some different mutation in the Regnase-1, which expressed the truncated Regnase-1. But even in this case, the Regnase-1 expression is really highly accumulated like this. So this means that in the UC patients, the frame shift mutation or loss of function mutation of the signaling molecules of the IL-17 signaling, but in addition, the gain of function mutation of Regnase-1 was observed. And the defect in the IL-17 signaling by this mutation leads to the survival advantage of these mutant cells and spread these mutations throughout the intestine. And that's why we found this mutation enriched in the UC patients. However, in the case of UC, there's a higher chance of the cancer progression, but the mutations were not in the colon cancer from the UC. So it means this mechanism looks to be totally different. So finally, we also try to identify the disease involvement of the Regnase-1 in the lung pathology. And as you can see, the Regnase-1 deficiency leads to the severe lung inflammation. And in that case, not only the immune cells like T cell, B cells, even airway epithelial cells are activated, and also the plasma cells. But we did not know what kind of cells are intrinsically controlled by Regnase-1. So we generated the bone marrow competitive transfer mice having the congenital with the different congenic markers of wild type and Regnase-1 knockout cells, and checked their the cells in the absence of Regnase-1. And we found that in the knockout, there's a huge accumulation of the ILC2 of the knockout cells. So ILC2 is a group to innate lymphoid cells, which function to highly produce type 2 cytokines such as IL-4, IL-5, and IL-13, although it does not have an antigen receptor. And it was known that the ILC2 is important for the immune responses against helminth infection and allergic inflammation. However, it's the role of ILC2 in non-allergic diseases are not well studied. So indeed, when we checked Regnase knockout mice, the numbers of ILC2 was highly increased in the knockout, even in the absence of T and B cells. And not only the numbers, the ILC2 activation marker ICOS is highly enhanced. And Th2 cytokines are highly produced by from the regnase knockout cells, knockout ILC2. So to further, so indicating that the regnase 1 is important for the suppression of the ILC2 activation. To further explore the mechanisms, we performed the RNA-seq analysis and discovered a set of genes upregulated in regnase knockout, including a set of type 2 cytokines. And when we checked globally about the diseases related with these genes, so we found, in addition to various general respiratory diseases, we found that pulmonary fibrosis is highly enriched. And indeed, when we transferred wild-type knockout ILC2 into the RAG2 common gamma knockout mice, which lack the ILCs in addition to T and B cells, and then induced the lung fibrosis by bleomycin. This is leading to the much more severe development of much more severe lung fibrosis when we transfer the regnase knockout ILC2s. So indicating that in mice, regnase 1 in ILC2 is important for the regulation or negative regulation of the lung fibrosis. So the question is, what is happening in human? So to check the clinical significance, we checked the regnase and protein expression in ILC2 in the bowel fluid from the idiopathic pulmonary fibrosis IPF patients. And I evaluated the relationship between the numbers of ILC2 and the clinical course of the IPF. And indeed, we found that there's a clear negative correlation between the regnase 1 expression and the ILC2 numbers in the valve. So it means that consistent with the fact that regnase 1 is important for the suppression of the ILC2 proliferation. And when we checked if there's a change in the ILC2 in the disease no progression or progression groups, so it tends to be the more numbers of ILC2 in the disease progressive IPF patients. So although it's not reached the statistical significance, because BAL-F, it's difficult to get so many patient samples, we switched from the BAL-F to the peripheral blood of the IPS patients and check the ILC2 numbers. So we hypothesized that more numbers of the ILC2 is leading to the poor prognosis of the ILC2. And indeed, when we cut off the ILC2 numbers as 1,500, the higher numbers of the ILC2 is really correlated with the much more poor prognosis of the IPF. So finally, this study indicates that the ILC2 is inducing the proteins like ICOS to induce its proliferation. And although I didn't show the data today, the GATA3 or EGF1, such kind of transcription factors to induce profibrotic cytokines to promote the pulmonary fibrosis. But regnase 1 acts as a key regulator of this ILC2 activation to suppress the human IPF progression. And although regnase 1 is important for the suppression of the inflammatory responses, but it can also suppress the anti-cancer immunity. So indeed, it was recently reported from a Humboldt-Cheese group that the regnase 1, if we mutate delete regnase 1 from CD8 T cells, the anti-cancer immunity was much more highly enhanced. So this could make sense. And finally, so in this talk, we found the importance of the regnase 1-mediated post-transcriptional regulation is important for the regulation of pathologies of inflammatory diseases, of pulmonary fibrosis, ulcerative colitis. but it also can negatively regulate the cancer immunity. But it is also possible that other autoimmune diseases or infectious diseases could be related to the post-trans B regulase 1 and post-transcriptional regulation. So which could be the further future direction of our study. And finally, this work was mainly done by postdoc Nakatsuka in collaboration with many collaborators. And thank you very much for your kind attention. I'll stop here and take some questions.
Thank you. Thank you very much for a wonderful talk. It's a kind of a lot of good data and new data for the audience as well as for me. So what we can do, we can take some questions at this point of time. So I will read a couple of questions from the audience.
Okay.
And, so one of the question is that length of poly A tail, whether the length of poly A tail determine the half-life of the mRNA. Would you give your opinion on this? So somebody is asking whether you have this mRNA having the poly A tail, and whether poly A tail length actually decide the long life of mRNA. I have no idea. So that's one question.
Yeah, that's a good question. In general, in the traditional dogma, when the poly tail is longer, that could be more longer half-life. But the recent mobile informatics studies rather suggest that, but this is not really the case. So the certain length of the poly A is more important for the control of the longer or more stable mRNA half-life. And also in terms of the regulation of the cytokine or inflammatory messenger RNAs, I rather don't think simply messenger RNA, the poly A length simply correlated with the mRNA, the stability of the mRNAs. So like as I explained, the endonucleases like regulase 1 is also important for the regulation of the messenger RNA in terms of the inflammatory responses. So it's not really simple.
Yeah.
And rather, of course, if the poly A is really short, this is kind of more intermediate of the mRNA, which are already undergoing its degradation. So in that case, the mRNA should be really gone soon.
Okay, so one more question we have is, can Regnase 1 could be used as a therapeutic target in COVID-19 patients, particularly to treat post-COVID-19 lung fibrosis?
Yeah, this is also a really good question. And actually, but not really about the COVID-19, but we are now trying to, we have some way to increase the Regnase 1 expression and even in vivo. And we are now trying to ameliorate the lung fibrosis. In this case, a more bleomycin-induced lung fibrosis model. But I think this type of the increase of the Regnase 1, based on my previous preliminary data, looks to decrease the severity of the fibrosis. And also, when we take advantage of that method, so we are still not using the COVID-19 itself, but for example, the ARDS model by the LPS intratracheal treatment. So that cytokine production by such kind of model can be suppressed by increasing the Regnase 1 expression. So I think the regulation to increase the regulation level is good to really nice to treat inflammatory diseases and also such as septic shock or ARDS type syndromes.
Okay, so thank you. If anybody in the panelist has a questions for Professor, if not, then I have a question. So basically, this is great data that you are showing that Regnase and ILC2 connection when you do this bone marrow chimera. But I could also see there is a upregulation or a frequency of basophils also increased in your model.
Yeah,
So
yeah,
yeah.
Yeah, thank you very much for that questions. In this time, we didn't focus on the basophils so much. So this is in part because this time we show the ratio between the control and knockout cells. But indeed, the frequency of the basophils are really low. I mean, only few basophils are present in the lung. So only a rather small difference also makes a really huge, fold change. So I rather thought that, but on the other hand, the ILC2 numbers are much more than basophils in the lung, at least. So I feel that it's more convincing to say that the Regnase 1 is controlling ILC2, but still the basophil control is, we are not really confident.
So because at some point of time there was a paper from Ruslan Madjitov lab talking about the role of basophil in initiating Th2 immune response, and since your ILC2 is also kind of Th2 cells, so somewhere is there any connection between the basophil and ILC2 in terms of Regnase 1?
That's also a good question. But so far we have not analyzed the basophils.
Okay.
But of course, there's a clear connection between Regnase ILC2 and eosinophils, although I didn't show the data. But in the case of basophils, still, we need to work a little bit more.
Okay. As I have one more question, then I'll ask one question from our panelists. So you also show, I mean, the other groups shown that the Regnase 1 and the anti-tumor immune response, right? So is this anti-tumor immune response somewhere Regnase is also regulating post-transcriptionally the checkpoint inhibitors, or it is through the cytokines?
So you mean that the regulation of the Regnase 1?
Whether Regnase 1 is regulating checkpoint inhibitors like PD1 or CTLA4 in terms of post-transcriptionally, or the phenotype what you see in the cancer settings is due to the hyper-cytokine response.
Yeah. So this is true. So really activation of these T cells is going to, so it's it's a bit complex. Even the PD1 itself could be regulated by the Regnase 1, but still, if in case the Regnase 1 is missing, the anti-tumor immunity is more enhanced. So even other effect overcome such a higher expression of the PD1.
Okay, so so one more question we have from Dr. Pawan Sharma. How much will H. pylori infection impact the Regnase 1 mediated pathophysiology? So he's trying to understand the connection between Regnase 1 and Helicobacter infection in terms of mediating pathophysiology.
This is interesting question, but so far we have no data. And but Helicobacter, you mean pylori infection?
Yes.
In the stomach.
Yes.
So
because it is also ultimately lead to some way like IBD, right?
Mm-hmm. Mm-hmm. Uh, that's an interesting point. So far, we do not have any good idea about this. But anyway, maybe I explained in the middle of the talk, the lignase 1 can be inactivated by the mode 1, which is found, whose mutation is found in the motoma, which is sometimes caused by the Helicobacter pylori infection. So it could be possible that in certain cases, the lymphocytes that are activated by this Helicobacter chronic infection might also cleave and inactivate lignase 1, and that can be also causing involved in chronic inflammation by the persistence of the Helicobacter pylori infection. But so far, I have no data, actual data about this.
Thank you. So I think this is a kind of one interesting question which audience is asking. One of the audience is, any studies on lignase 1 mutations have been associated with the Crohn's disease patient?
Crohn's diseases. So far, there's no report about this.
Or
That's an interesting question.
let's put it another way. Is there any other kind of GWAS correlation between lignase and IBD in terms of either UC or Crohn's?
In that
Genetic
case, no.
association.
And even in our case, that mutation we really didn't expect. So maybe one reason is in the case of GWAS, this is more related with the let's say more but at least in our case, the mutation is really rather gain of functions, and the most host spot is really specific to the phosphocytes. That was really surprising for us. But so that why maybe by more GBAS type marker type screening, it could be really difficult to be identified. So this is my guess. So
All right. Sounds good. Then I don't think we have much more question at this point of time. Uh, Thank you very, very much for wonderful talk, Osamu. And
thank you.
now I'll take this opportunity to introduce our next speaker of this session. And she is from PGI Chandigarh, uh, Professor Gupta Sharma. She is going to talk about the MHC linked control immune responses. And basically, she has Professor Mani Luthra Gupta Sharma has a bachelor degree in chemistry from Delhi University, a master degree in biotechnology from IIT Mumbai, a PhD in molecular biology from CCMB Hyderabad, and postdoctoral training he has done from University of Cambridge. I always go to Oxford, so it's always confused about Oxford and Cambridge. So University of Cambridge. She has worked as a faculty in Punjab University and at PGI Chandigarh, where she is presently a professor at the Department of Immunopathology, conducting supervising research in several different areas of immunology and ophthalmology. Her area of expertise, in addition to above the molecular and cellular immunology and cell biology, her current research interest includes elucidation of mechanisms and pathways involved in immunopathological diseases such as ankylosing spondylitis, rheumatoid arthritis, as well as ocular disease that involve like fibrosis in the cornea, lens, and retina. She uses her background in protein chemistry, as she's a trained protein chemist, and protein engineering, using fast display technology to create novel tools to study some of these mechanisms and pathways. So she's going to talk about MHC system, and without wasting time, I would like to invite Professor Gupta Sarma to please deliver her lecture. Thank you.
Dr. Mani, are you around? Can you start your presentation, please? Unmute yourself, Dr. Mani.
I can see some activities.
She's trying to find out how. Yeah.
I request everybody to please mute yourself because we are getting some noise in the background. So please mute yourself so that there is a clarity in the presentation. Thank you.
Can you see my screen now?
Yes, we do.
Okay. Sorry for this delay. I'm extremely thankful to the organizers for giving me this opportunity to present a lecture in this prestigious course. And I'll be talking about the major histocompatibility antigens. And I was told to keep this as a very basic talk, chiefly directed towards the students, which I will be doing. So major histocompatibility antigens are basically found on the surfaces of nearly all human cells, and they are glycoproteins in nature. They were discovered several years ago by George Snell, who was trying to do some transplantation experiments in mice. And he found that while some tissues from donor mice were accepted by the recipient mouse, other tissues from other donor mice failed to be accepted and were rejected. When he tried to find out what is the reason behind this, he noticed that if the animals were not closely related, the recipients rejected the graft. But if the animals had a similar genetic makeup, then the donor graft was accepted by the recipient. So these genes, which are responsible for this kind of acceptance of the graft, were later called as the major histocompatibility antigens, which are encoded for by genes which are present within the major histocompatibility complex. Now, histo means tissue and compatibility means getting along. So these genes were named for their association with histocompatibility, and it was called as a complex because these genes belong to a very large genetic region which contain multiple loci. So together it was known as the histocompatibility complex. Now the prefix major was included to this complex because all these proteins which are encoded by these genes were found to have very dramatic effects on histocompatibility. And also it was useful to include the term major here to be able to distinguish them from some other proteins which lie outside this loci and which have minor effects on the histocompatibility. So basically, the major histocompatibility complex gene products are directly involved in immune responses. This is a picture to show you the genomic landscape of the MHC, which comprises about 260 genes spanning about 4 MB region on the chromosome short arm of chromosome 6p. And just to give you an idea of how big this chromosomal region is, this base 4 MB region is approximately the size of the entire genome of the E. coli. So it's a very huge region, And that is why it is also known as the complex of these genes. So another thing that I would like to point out here is that the portions shown here in yellow are the ones which include the genes belonging to the class 1 region. The MHC class 2 genes are present in this blue region, and the MHC class 3 genes are present in this region, which is shown here in pink. So the MHC class 1 genes are actually the ones which present the antigen to the cytotoxic T lymphocytes or the CD8 plus T cells, and the antigens which it presents are basically to eliminate the viruses. So these are virus infected cells or the tumor cells, whereas the MHC class 2 antigens that are encoded by the MHC class 2 genes express and present peptides to the CD4 T helper cells, and therefore they are very important in the initiation of cellular as well as humoral immune responses. The MHC class 3 products are only producing secreted proteins, but have very important immune functions. For example, proteins belonging to the complement system, as well as the inflammatory molecules. So the gene organization in humans is found in chromosome 6, and the counterpart in mice is found in chromosome number 17. In humans, it is also known as the human leukocyte antigen or the HLA complex, whereas in mice, it is known as the H2 complex. Now, as far as the expression goes, the MHC class 2, unlike the MHC class 1, which is expressed constitutively on all nucleated cells of the body, the MHC class 2 constitutive expression happens only on the antigen presenting cells, that is the B cells, macrophages, dendritic cells, as well as the epithelial cells of the thymus. But the expression of MHC class 2 can be induced in the presence of cytokines like interferon gamma or upon activation of the T cells. So as I said, the expression of MHC can be regulated by cytokines. And then there are various transcription factors that help to increase the MHC gene expression. But there are some factors which might decrease the MHC expression or even include the removal of the total loss of MHC expression, and those are the viruses. So some viruses help in decreasing the MHC expression, help themselves to evade the immune response from the host. So this is an overview of the various elements that are present in the MHC, upstream of the MHC. So in all cases of the HLA class 1 molecules, except the HLA-G, there are some promoter elements like the enhancer A, the interferon stimulatory response element, and the SXY module, which are the promoters which bind to transcription factors like the NF-kappa B and the interferon regulatory factor 1, which bind to these and then activate the TNF alpha in the interferon gamma signaling pathways and result in the increased expression of the HLA molecules. The important point to note here is that all these sequences contained within these promoter elements vary a lot between one HLA to the other. And it is these variant sequences which are present in the promoter region which dictate the level of promoter activation and thereby consequently dictate the level of expression of the MHC genes or proteins. So as I said earlier, the MHC class 1 molecules present antigen to the CD8 cytotoxic T lymphocytes, whereas the MHC class 2 molecules present the antigens to the CD4 T helper cells. Other than the T cells, the MHC molecules also have receptors on the NK cells, like both the activatory as well as the inhibitory receptors present on the NK cells, as well as the leukocyte immunoglobulin-like receptors present on cells of the myeloid monocyte lineage. Now, included within the MHC complex are also the class 1b genes and the class 2b genes. So these are the non-classical MHC genes and present within the same area where the class 1 genes and the class 2 genes are present in the MHC region. The class 1b genes encode for proteins like HLA-E and F, which activate the gamma delta T cells. The HLA-G which is a very important molecule and expressed on the cells of the placenta. And these are responsible for the inhibition of the maternal immune response, which could happen against the fetus. The HFE, which is involved in iron absorption in the cell, and the MIC-A and the MIC-B, which are also like the class 1 molecules in structure, but they are expressed in terms as stress molecules. Now, the class 2B molecules have a structure very similar to that of the class 2. Class 2B have a structure very similar to like the conventional class 2 molecules. And these include the HLA-DM, which has an important role in the antigen loading onto the class 2 molecules, and the HLA-DO, which suppresses the action of the HLA-DM. Other than this, there are some MHC-like genes which encode for proteins which lie outside the MHC region, but their structure is very similar to that of the MHC in combination with beta-2M. And these antigens actually have peptide binding regions which are very hydrophobic. And as a consequence of that, these, like the CD1, binds to lipids and glycolipids. So let's look at the overall structure of the MHC class 1 molecule. It's made up of 2 chains, one of which is the alpha chain, also known as the heavy chain, and consists of the alpha 1, alpha 2, and alpha 3 domains in its extracellular region, continuing into the transmembrane region and then into the cytoplasmic domain. The alpha 2 and alpha 3 consist of an intra-domain disulfide linkage, and the alpha 3 domain is non-covalently associated with a small molecule about 12 kDa in size called the beta-2 microglobulin. And the presence of beta-2 microglobulin with the alpha 3 non-covalently associated is very important to stabilize the structure of the class 1 molecule. Together, the alpha 1 and alpha 2 domains make up the conformation which presents the peptide to the CD8 T lymphocyte. So here is the alpha 1 and alpha 2 domains and binding to the peptide. And it can be seen that the binding of the peptide happens in the peptide binding pocket, which is provided for by 2 alpha helices. And the floor of this peptide binding pocket or cleft is made up of beta sheets. Now, if we look at the structure of the class 2 molecule, it is a heterodimer consisting of an alpha chain and a beta chain, and both are made up of 2 domains each, the alpha 1 and alpha 2, and the beta 1 and beta 2 domains in the extracellular region, followed by the transmembrane domain and a cytoplasmic domain. The peptide binding cleft is formed by the joint cleft provided by the conformation provided by the alpha 1 and the beta 1 domains of the 2 chains. There is no beta-2 microglobulin in the class 2 molecules. So here is the peptide, which is shown here in green, binding to the cleft provided for by the alpha chain as well as the beta chain in combination. If you look at the cleft geometry, there are some differences between the clefts in the MHC class 1 molecule and the class 2 molecule. In the class 1 molecule, the cleft is more deep and narrow, whereas in the class 2 MHC molecules, the cleft is more broad and shallow, as a result of which the class 1 MHC can accommodate smaller peptides of 8 to 10 amino acids in length, whereas the MHC class 2 binds to peptides which can be more than 13 amino acids in length. So a large number of studies have been done by eluting out the peptides from the MHCs in order to be able to determine what are the kinds of sequences that bind to the MHC class 1 and class 2 molecules. And such detailed studies have resulted in some classical consensus sequences, also known as motifs, from where one can find that sequences towards the N and C terminus of these small peptides which bind to MHC class 1 molecules are rather conserved and have either hydrophobic residues or aromatic residues on their N and C terminus. These residues are important because they act as anchors to bind this peptide within the groove or the clefts of the MHC class 1 molecule, and those clefts or where these anchor residues accommodate themselves are known as the pockets of the MHC molecule. Similarly, similar studies were done with MHC class 2 peptides, and it has been found that there are no anchor residues on the ends of these chains or towards the N and C terminus, and the motifs are less clear than in the class 1 binding proteins, but the pockets are definitely more permissive and more flexible in nature. Now coming to the concept of polygenicity of the MHC class 1 and class 2 genes. Normally in a human body, you would find that one gene encodes for a unique protein and that has a unique function. But in the case of MHC, this is not ideally so. That is to say that there are multiple genes, all of which can code for the same kind of MHC molecules, resulting in the same kind of functions that these will lead to. For example, the HLA A, B, and C of the MHC class 1 can all give rise to the alpha chain of A, B, and C respectively. And each of these can then combine with the beta 2N to give rise to the HLA A, C, and B, all of which provide similar functions. Similarly, the HLA DP, DQ, and DR present within the MHC class 2 chains can give rise to the alpha and beta chains of the DP, and these combine to give rise to the HLA DP. But in case of HLA DR, there is only one alpha chain, but there are up to 9 beta chains possible. And so this alpha chain can combine with DR beta 1, beta 2, and so on to give rise to multiple combinations of proteins. However, it has been seen that only DRB1, 3, 4, and 5 actually give result to a functional protein. So no two individuals might have the same number of HLA-DR chains present in them. So this is the concept of polygenicity, and this is one reason why two individuals may be different from each other. The other reason why they are different, and a very important reason, is the polymorphism of the MHC genes. These genes are the most polymorphic genes known to date, and it was Haldane in 1949 who proposed the role of infection in driving polymorphism of genes. And so these MHC genes have evolved over time to be highly diverse in nature through mutation and gene duplication and conversion, so that the population eventually has a very diverse allele combinations of HLA class 1 and class 2, leading to herd resistance to infection and a survival advantage. Contrast this to the cheetah. The present day cheetah is thought to have arisen from a limited breeding stock, causing a loss of MHC diversity. That is why cheetahs are very highly susceptible to viral infections, because it has been proposed that the original population itself was a very limited breeding stock. And so you can see that the greater the polymorphism or greater the diversity, greater is the resistance to infection and survival in the population. Now, it would be expected, and it is naturally so, that most of the polymorphic residues of the MHC molecules are located in the peptide binding sites of the molecules themselves. So you could see that present here in different colors. And in the HLA-DR, the variations are more in the beta chains. The alpha chains are non-polymorphic. To date, these are the numbers of the HLA class 1 and class 2 alleles that have been found, and these are continuously updated on this site. And you can see that all this polymorphism in the MHC loci has arisen over time, either because of gene duplication, deletion, gene conversion, or allele conversion. So the diversity in the population has arisen because of polymorphism, but even in an individual, there are multiple steps which can result in diversity. As we saw, one is polygyny, the other is polymorphisms, and the third reason is that the genes in the MHC are very tightly linked, and they are usually inherited in one unit called the haplotype. So this is one haplotype that a person has inherited from the mother, let's say, and this is the other haplotype which has been inherited from the father. And further, each of these products of these genes are co-dominant, that is, they are equally expressed. So there are 6 of these HLA class 1 genes and 6 of these HLA class 2 genes which are expressed in the individual, making this individual different and unique. When this combination of haplotypes is passed on to the progeny, then the inheritance happens in a Mendelian fashion. So one would expect that 50% of the siblings would be a haplomatch or 50% match with each other, and 1/4 would be actually non-matched with each other or totally unmatched, and you could expect 1 in 4 to be totally matched with each other. A very brief mention of how the antigen processing takes place in both cases. In the first case, that is where the MHC class 1 antigen presentation is happening, the endogenous proteins are degraded in the proteasome, in the cytosol, and these peptides are led into the endoplasmic reticulum through the transporter associated with antigen processing, where these peptides are loaded onto the MHC. Now, the MHC heavy chain is assembled along with the beta-2 microglobulin, along with associated with various chaperones who help in the process, and the peptide is loaded onto the MHC class 1 molecule, from where it proceeds to the Golgi apparatus and then to the cell membrane, where it presents the antigen to the cytotoxic T cell. In the case of the MHC class 2 antigen presentation pathway, which involves the taking up of the exogenous antigen, the exogenous antigen is taken up by endosomes, then that leads from the early endosome to the late endosome to the lysosome, where the protein is chopped into multiple peptides, and the MHC, having been started off from the rough endoplasmic reticulum, where the cleft is actually occupied by an invariant chain. This invariant chain is proteolized in the presence of a low pH to result in the formation of a clip, which helps to keep this peptide binding pocket occupied until it meets the necessary peptides to be loaded, where the clip is exchanged for by the peptide, which has to be presented on this MHC with the help of the HLA-DM molecule. And then this MHC peptide complex moves to the cell membrane, where it is presented to the CD4 T cell. It is very important that this interaction takes place in a programmed manner, that is the T cell receptor with the peptide MHC in complex. And this part was discovered long back when in the 1970s. In fact, in 1970, neither the T cell receptor nor the peptide MHC complex surface were worked out. In 1973, both Peter Doherty and Zinkernagel were doing experiments with looking at the T cell responses to the LCMV virus. And they found that the cytotoxic T lymphocytes from the virus-infected mice could only kill infected cells derived from inbred strains of mice that had the same MHC haplotype. What this means is that if there is a T cell of an individual who is HLA-B8 positive, then this T cell will recognize the cognate antigen only in the context of the antigen-presenting cell, which is from a HLA-B8 background. But it will not recognize the antigen presented by HLA-B44 and will not recognize a non-cognate antigen, even if presented on an HLA-B8 background. So this part was dealt with in the morning by Dr. Nandi in detail, and I'll just skip over it very quickly, saying that these thymocytes are trained in the thymus by a selection process, the first of which includes apoptosis of most of the cells that do not interact with any HLA molecules, and followed by a positive selection of thymocytes whose receptor bind to the self-MHC molecules. Having selected a large percentage of these cells, a large number of these cells undergo another step of selection, which is known as a negative selection, whereby all these thymocytes, which have very high affinity receptors for self-MHC and self-antigen, actually undergo apoptosis. So more than 95% of the cells have finished in the thymus, having been negatively selected for, and the only cells that actually exit the thymus are the single positive cells, which exhibit specificity for self-MHC and foreign antigen, but lack autoreactive properties. So there is a very great importance attached to the HLA peptide and the TCR recognition. And other than the conventional docking there are alternative mechanisms which can affect this kind of interaction, and this interaction can go haywire and result in disease. And some of these mechanisms are alternative docking, PCR stabilization of weak peptide HLA complexes, altered register, hotspot binding and molecular mimicry, post-translational modified based modification, altered HLA binding or antigen processing, differential stability of HLA molecules, or neoantigen presentation by the peptides, all of which can lead to the downstream consequence of increased TCR-mediated activation of autoreactive effector T cells. Further, this threshold of activation of autoreactive T cells can be modulated also by non-HLA risk genes and environmental influences. So it is thought that this avidity and affinity of the peptide MHC TCR interaction could dictate whether the MHC would be a protective MHC or a risk-bearing MHC. If there is low affinity avidity of this interaction, then there could be defective negative selection and autoimmunity. Whereas if the interaction is intermediate or high, then it can result in enhanced negative selection and agonist selection of the T regulatory cells, resulting in dampening of the autoimmune response. So some experiments have been done where it has been shown that if there is autoreactive thymocytes and then there'll be failure of the negative selection and there is a lack of any protective MHC molecule. Whereas if there is transgenic expression of a protective MHC molecule in the dendritic cells, then it can lead to effective negative selection and clonal energy of these autoreactive T cells, also development and differentiation of the T regulatory cells, which can then leave the thymus and enter the draining lymph node, where they can suppress the activation of these autoreactive T cells and by directly acting on the antigen-loaded dendritic cells. So it is thought that the MHC class 2 molecules have actually evolved based on their ability to clear different kinds of infections. For example, interferon gamma is very important in clearance of viral and intracellular bacterial infection, and HLA-DQ6 is known to promote the activation generation of interferon gamma from the CD4 T cells. Similarly, IL-17 is important in the clearance of extracellular bacterial and fungal infections, and HLA-DQ8 is efficient in the generation of IL-17 from the T17 cells. So in the case of HLA-DR, they are able to take care of both kinds of infections because of moderate amounts of production of both interferon gamma and IL-17. So it is thought that these three classical HLA-2 haplotypes have survived the bottlenecks of infectious episodes in the history of humankind because they have been able to help in the clearance of different kinds of infections. But at the same time, some of these class 2 molecules are very autoimmune prone because of the generation of or secretion of pro-inflammatory cytokines. And some of the most important autoimmune diseases like RA, multiple sclerosis, SLE, type 1 diabetes are known to be associated with the presence of these class 2 molecules. So if there is a particular combination of a heterozygous haplotype which involves this and that, then there'll be exaggerated amount of pro-inflammatory cytokines that are produced, and this would predispose the individual to autoimmune disease. On the same lines, there could be protection from autoimmune disease if there are anti-inflammatory cytokines resulting from the interaction of particular MHC class 2 haplotypes. And this is what is found for HLA-DR4, which is protective against RA because of the production of TGF-beta and IL-10, and HLA-DQ6, which is responsible or seen to be protective for multiple sclerosis. Now, DQ6 is an autoimmune resistant haplotype, which produces interferon gamma, and it also helps to modulate the amount of IL-17 produced by the HLA-DR2 if it is present as a single haplotype. So it helps to actually cause resistance to autoimmunity. Now, this is an example of a trinity of HLA-B alleles in HIV-1 infection, which I present as an example. Here, both HLA-B27 and B57 are known to be protective towards HIV infection, whereas HLA-B35 predisposes the individual to HIV-1 infection. It is thought that these two alleles, these two antigens, that is B27 and B57, might actually gradually decline. And that is because the virus is continuously trying to generate escape mutants to be able to escape the immune landscape of the host. So this is how mutations gradually take place in the pathogen to be able to escape the immune landscape. And this is a summary of all the HLA diseases that are known associated with the MHC class 1 and class 2 antigens. And this is a slide which I've left to just leave you with it, showing that our lab is also working on the structural biochemistry of HLA-B27 association with ankylosing spondylitis. This is the association which is very strongly proven, and we have presented a hypothesis for it. We think that the misfolding of HLA-B27 into high molecular weight aggregates results in the ankylosing spondylitis, and we are working on different subtypes of it. Interestingly, different subtypes of the same molecule HLA-B27 may be either associated with disease or non-associated with disease. And we are looking at the different clearance mechanisms of the high molecular weight aggregates of HLA-B27 among the different subtypes of it, whether they are disease associated or non-associated. Thank you for your attention.
Thank you, Professor Gupta Sharma. Wonderful talk. And we have some questions from the audience. Let me read some of these. So one of the things is whether monozygotic human twins have the same histocompatibility molecules on their cells. Can they accept transplant of tissue from each other? That's one question.
So I would think that there is a greater chance of the homozygotic twins to be able to accept transplants, although in any case, in none of the cases, the one is completely foolproof safe, because there are several minor antigens also which could have a effect on transplantation. And so it is in totality that one has to see the effect of the graft on the host.
Okay. Another interesting question is whether what is the possibility of high affinity MSC class 1 or class 2 restricted protective peptide for mouse, say for influenza, would also be protective in human? So basically, can I repeat the question?
Yeah, I just, I'm trying to understand what the question is.
Somebody is, S. Kumar, trying to ask you a question whether if you find a protective peptide, whether it's a class 1 or class 2 restricted in the mouse, yeah, whether the same peptide can be used as a protective one for humans. Let's say, for example, influenza.
Oh, okay.
Peptide. Yeah.
So actually, there are several homologies that are possible between the mouse and the human, and there are umpteen number of softwares that are available in which one can feed these peptides in the context of the MHC that one is trying to work upon to see whether the MHC will be able to bind to that peptide or not. So these are bioinformatic tools which one can use to see whether a fit happens or not. And by and large, these tools are very useful and effective also, because if these can be used in actual synthesis of peptides, then it has been seen that they give good results. So there is quite a lot of compatibility between the software and the actual experiments.
Okay. So there's a possibility, but you have to have a prediction and software. Yes. So another one is when an individual has to undergo an organ transplant, to what extent should the MSC match? I mean,
so as far as solid organ transplant is concerned, even a 50% match is okay, or even actually sometimes the match is not determined before the transplant. So one can still go forward for the transplant, but by just looking at the cross-reactive antibodies. So serological testing is done to look at the antibodies, but tissue typing one need not do, because in any case one goes through a lot of immune suppressants after the transplant. But this is not so in the case of a bone marrow transplant, where ideally one tries to look for a full match. And if a full match is not available in the immediate family, which can only be possible among siblings, because you will only get a haplo match if you looked at the parents, then one has to go into the donor registries and look for a full match.
That sounds good. So if anybody has a question from the panelist, if not.
May I ask a question, please, Amit?
Yeah, please go ahead.
Yes, Dr. Mani Luthra, it was a very, very lucid and clear presentation. I have a quick question. You mentioned that aggregation of some of these HLA molecules, such as B27, which you yourself are studying, could be predisposing or not predisposing to a disease. Has such association been shown so far in any particular disease?
So in the case of HLA-B27, it has been thought that disulfide-linked dimers of HLA-B27 can cause disease. And we have ourselves shown that aggregates of HLA-B27, which need not be disulfide-linked but non-covalently associated aggregates, can also cause disease. And this we have shown by actually making antibodies in our lab and looking at the patient samples themselves. So it is not as though these high molecular weight aggregates are aggregates per se in the particulate form. These are soluble aggregates. So they are very large molecular weight aggregates which are soluble in nature. And we believe that these are the large aggregates which can result in an immune response because they are seen as non-self by the immune system.
Okay, so you mean are they also seen in ankylosing spondylitis, where people are very highly predisposed, having been associated with HLA-B27?
So nobody else has ever worked on the high molecular weight aggregates. People generally tend to ignore them because they think that these are not things to be worked upon. But so we are the only people who proposed it, and we have shown that these aggregates do exist in patients, because we made antibodies which are cognate to the high molecular weight aggregates and do not bind to the complex, ternary complex of the HLA-B27. And we showed their existence in the patients, as well as tried to distinguish the HLA-B27 positive individuals who have not yet developed disease versus HLA-B27 individuals who are patients who have ankylosing spondylitis. So with those experiments, we have been able to prove our point that HMW aggregates are important in the context of AS at least.
Thank you very much. Also, it makes me wonder whether such predisposition to form soluble aggregates would vary in individuals with different ethnicity. Reason being that it is not, maybe all individuals who have HLA-B27 may not be predisposed. So would it also, although you may not have studied it, so would you, be able to comment on this, that whether it would vary with the ethnicity? Dr. Mani, was I audible? Was my question audible?
I'm not so sure. She got disconnected.
Yeah, question was clear and loud, but I don't know where money is gone.
I think there's
there are a couple of questions in the new questions. Can you ask those questions in the question answer box? You can check.
You're talking to me?
Yeah, there are a couple of more questions in the question.
I know, but I think somehow she is disconnected, I guess. Hello, is Professor Gupta Sharma, are you there online? I can't see her in the,
Dr. Mani is not there. I think she left the session.
Yeah. So,
okay, we can close the session.
So I think it's great. So we had really a wonderful session with the two speakers, and both really spoke in a depth. as well as in a teaching mode, so that the student can also got benefited. So now we are having a 20-minute break, and then we will be starting another session, and Dr. Dil Afroz will be moderating that session. So let's get back in 20 minutes. Thank you.
So, guys, we have a short break. You guys can have tea or coffee, and we'll be back in 20 minutes. Thank you. Hi, Dr. Dilafroz. How are you?
I'm good.
Very nice. Good to see you. So,
yeah, but you aren't able to see me yet.
Pardon me, you can?
I said you can only hear me, you can't see me. So you have to allow me to start the video.
You should. You, as a panelist,
So,
you have
it says that you can't start your video because the host has stopped it.
No, you try it, you will be allowed.
Okay, maybe now I am allowed. Now I'm allowed. Oh, it says it's again. I think we have to ask Gul to allow me to start it.
Ma'am, we have taken a break for 10 minutes. After that, only we will start the session.
Okay.
Yes, ma'am.
So,
but I want to talk to Dilafroz, so ask Anupam to kindly allow her.
Yes, I'll ask him to do that.
yeah, there I can now.
Great.
We can see.
Hi, sir. How are you doing?
I'm good. How are you?
I'm good too, sir.
But I can't see you again.
Still you can see me, but I can see you.
Now it is all right. Yeah, this is good now.
You can see me now? Okay.
Yes.
Great. How are you doing, sir?
I'm good. आप बताइए कैसे हैं? How is Srinagar?
Oh yeah, we are. Today is warm, but otherwise we were like down with temperatures for the past few days. It's great. It's been quite a stimulating session this morning. Wonderful talks and all.
Dr. Mani, we missed a couple of, Dr. Mani, we missed a few questions. You were offline quickly.
That's right.
What happened?
Suddenly my Geo internet connectivity went off, so I reconnected.
No problem. It was a wonderful talk. Yes. I really enjoyed. I have heard so many times, but this was so nice. It clarified so many, doubts in my mind. Thank you for giving such a lucid talk. Very nice.
Thank you for giving me this opportunity. I'm really thankful to you.
We'll call you once more because now we have chosen you as one of the very nice people. Very nice.
Thank you. Thank you.